Good morning to everyone. Good afternoon, depending on what part of the world you're in. Welcome to our second day of the virtual symposium in plant omics science. My name is Andres Jaramillo Botero. I'm the Omics Program Scientific Director, and I will be chairing the session for today. We appreciate your presence, special guests, researchers, students from the different institutions that have registered in the program. And we're looking forward to a very productive session this morning. Please bear in mind that some of the presentations that you will hear in this session are in Spanish and others will be in English. In any case, we will have our questions and answers session for every presentation, and we look forward to your interventions. Our first speaker today is going to be Juan Manuel Marmolejo. Juan is an electronic engineer with a doctorate in engineering, emphasizing in semiconductor design, or basically nanoscale semiconducting devices. He will be talking today about electronic transport and nanostructured materials and their use in nanosensing or biosensing or sensing of biomarkers, which is very relevant to our interest in phenotyping of plants. Juan, good morning, and please proceed. Thank you.
Our most recent results in graphene nanoribbon-based devices for the indirect detection and measurement of sucrose that can be useful for monitoring the stress response of plants. I will begin with a short motivation, followed by an introduction on field effect devices, graphene and 2D materials in general, and our computational design and characterization of two kinds of biosensors based on graphene nanoribbons. We all know the importance of detecting and measuring specific molecules in living organisms for different purposes. For instance, these molecules could be indicators of a disease or could help to determine the response to various stresses, effectiveness of treatments, and many others. If we begin from the extraction of some fluid in the organism, its traditional analysis consists on sending these samples to a remote laboratory, which implies high cost and time requirements. The objective of modern biosensors is to enable portable, low-cost, and easy-to-use systems for analyzing the samples on site and avoiding long-time transportation and reading of results, for which smart devices could be employed and diagnosis could be done within minutes. On the left, we have a traditional field effect transistor with an N-type channel. Under this zero bias condition, electron transport through the channel is negligible. However, when we apply a positive gate voltage, we get accumulation of negative charge along the channel, enabling a passage for electron transport. To the right, we see a characteristic IV plot for this device, where we can check that current flux is modulated through our gate voltage. On a field effect-based sensor, we look for current modulation through the absorption of molecules on the channel of the device, having a similar effect to that of a gate voltage, so that different concentrations yield to differentiable changes in the electronic properties of the device, as we see on the figure to the right. By doing so, it is possible to detect and measure target molecules with very high precision and sensitivity. 2D materials, which are single or few atom thick crystalline structures, are particularly interesting for sensing applications because they have a very high surface to volume ratio, among many other properties. We can have single crystals or combinations of two or more atoms, as we see in the figures, but we will concentrate on graphene that is formed by carbon atoms in the honeycomb lattice that we see on top. Large area graphene is metallic, meaning that it is not possible to simply use it as a semiconducting channel of a typical field effect device. However, there are some well-known strategies for opening the band gap of this material. One of them is cutting the system into very narrow stripes that are known as graphene nanoribbons or GNRs. whose edge defines their name and electronic properties. Then we can have armchair or zigzag edge nanoribbons that have all been confirmed to be semiconducting experimentally. However, zigzag and specific family of armchair GNRs known as 3M plus 2 or alpha as in the figure have the smallest gap, and this will be important for the sensor designs and characterization that follows. Our first design consisted on an all-armchair GNR field effect device where we use a semiconducting channel and near metallic electrodes. We functionalize the channel with a self-assembled monolayer of pyributyric acid linkers that non-covalently bond to graphene surface on one end and enables binding of target UDP glucose molecules to the other end. This molecule is particularly interesting because it is an intermediate reactant in the synthesis of sucrose and an indirect and proportional indicator of sucrose production within a plant cell cytoplasm. As seen on the figure to the right, we get a reduction in the current response of the device as the concentration of UDP glucose increases. Then this device is highly sensitive to UDP glucose and is useful for single molecule detection and measurement with detection limits in the picomolar range. Furthermore, the device could be functionalized with other kinds of linkers for the selective detection of different target analytes. However, these kind of devices rely on the fabrication of GNRs with very well-defined edges and low defects that are still difficult to achieve. Then, our next design aims to overcome this issue by using commercially available large area graphene devices that have metallic behavior. Here is our multi-terminal device that uses near metallic channel and electrodes. The channel is also functionalized with pyributyric acid linkers, and we apply a voltage difference on the left electrode with respect to the other electrodes and measure the current flow to each of them. By increasing the concentration of UDP glucose molecules on the channel, we found that the current flow from the left electrode to the lower electrode was decreased, as observed in the figure. We also try to increase the width of the channel in order to have a smaller band gap, where we can still see some difference in current flow as more UDP glucose molecules are added. But we still need to confirm this behavior when using a fully metallic system. We observe that the addition of target analytes in the channel proportionally reduces the density of states projected on the lower electrode, which then causes current flow to be decreased as we previously saw. Therefore, our preliminary results show that single molecule detection is also possible with this multi-terminal near metallic setup. Then we have the graphene nanoribbon devices are suitable for sensing applications where we expect to have very high accuracy, stability, sensitivity, and selectivity capabilities than conventional approaches. Although we know the difficulties in obtaining defect-free GNR structures, we have proposed some workarounds, such as employing commercially available large area graphene structures with metallic properties. However, we need further studies to generalize the behavior of our device and to confirm its sensing capabilities experimentally. Lastly, the presented designs could be architectural nodes for more complex applications where we can have very selective sensors that may be later embedded in mobile devices and so on for a wide range of applications. We thank you for your attention and look forward for possible questions you may have.
Okay, Juan, thank you for your talk. I believe by mistake you made a shorter video, so if you want to add anything online, please do so.
Okay, so thank you so much. Yeah, by the way, I prepared an 8-minute video, but okay, we can extend it a little bit more. Basically, I wanted to say that we're trying to, sorry. Basically, we're trying to make these devices experimentally. So one of the ideas is to use commercially available fully metallic graphene devices. So that was related to our latest approach. We're still working on this, and we hope to work on functionalizing these kind of materials and then trying to make some measurements. Another idea we're trying to explore is using using reduced graphene oxide, so that we can have some kind of semiconducting channel in our device. And we will try to make some first in silico designs to functionalize these devices, see how stable are these structures, and also try to find experimentally if we can measure indirectly sucrose or any other kind of molecules that are related to measuring the stress on plants.
Okay, so Juan, let me start with some questions online. Professor Luis Eduardo Tobon is asking, I mean, he's first congratulating you. And then he's asking, what kind of external circuit does the device require for real applications? And is it useful for in vivo measurements? And I will add to that, how do you think this would work in a solvated environment? And in particular, how would you compensate for a probable low signal to noise ratio?
Okay, yeah, very interesting. I invite you to look at one of the sessions we have later on from Sami in our group, working basically on how to measure, how to take measurements of these devices. But basically, we need some sort of deal to try to minimize all the noise that can come from thermal issues. But the good thing about these devices is that graphene is very stable under solvent conditions. So the idea of this is that you can have some solvent on top of the channel and you can still make measurements. We will have to try to amplify the signal as best as we can, so that we can have single molecule detection. But I don't know all the details about this experimental approach. And hopefully we can have a better idea later on when we see some approaches that we're working in the group.
Okay, so part of the idea of having ligands, and you were using pyrene butyric acid ligands to the graphene, which are non-chemically bound to the graphene surface through aromatic or through pi stacking. Do you think that will be relevant in terms of reducing the thermal noise in your system?
Yes, of course, because these molecules are very sensitive to measuring. In this case, they are reactive to one of the ends of UDP glucose, which is part of sucrose. So we expect that by using specific ligands on top of the channel, we can reduce this kind of thermal noise because we can have molecules adding to the surface of graphene. But if you're able to fully stack this pyrene butyric acid on top of the channel, we could reduce this kind of noise.
Sure, but the pyrene butyric acid is floating on your graphene, so it's limited or restrained on the Z direction. That's normal to the plane of the graphene, but it's not restrained on the others. So
yeah.
Do you think there's going to be any transition in your electronic signature as the pyrene butyric acid just shifts from pi bond to a different one in the graphene surface?
Well, it shouldn't, because when we made the calculations, we placed this pyrene butyric acid randomly, and we saw an increasing, like a proportional difference in the signal that we were measuring. So it shouldn't be a problem. But yeah, we also found in molecular dynamics calculations that they float on top of graphene. So it will be a challenge to just try to stack them in a way that they follow as minimum as possible. But we also saw that if we put too much, like a very big concentration, they will try to stack on top of each other. So that would be a problem. Also, it's likely that if we put a very high concentration, they will stack vertically one to each other. So that's some of the challenges that we will have to deal with these kind of designs.
Okay, so I think Luis Eduardo has another suggestion, and I believe he's worried about how to actually extract the signal. And in these devices, we're extracting the signal directly as an electronic signature. Basically, it's a switching transistor device that's switched by the molecules. So Luis Eduardo, you want to ask your question on the mic? Should I open the mic for you, or let me see?
Yeah, I'm here.
Okay, go ahead.
Then my opinion is that, my idea is that we can change maybe the capacitor or a capacitance, an internal capacitance in the device. Then we can connect this device to a radio frequency transmitter. Then we can use this resonance to check in an external device the concentration of the sucrose or another element you want. It's just like a resonator inside
Sure.
the plant or the animal or whatever.
Sure, so that's more like a piezo material in which you have the absorbed mass of the molecule change your resonant frequency, right?
No, it's not mechanical
really.
system. It's a electronic system. Then,
but
maybe the molecule
extracting the capacitance might be hard in the graphene devices, because there is really, there's a dielectric, because Juan has showed he has a back-gated device. And extracting the capacitance there might be a bit of a challenge, but I understand your suggestion.
Yeah,
Juan,
but if the channel change, then the internal capacitance change. No, it's not like
sure.
Yeah, actually, I have seen a few approaches similar to that, where they have some graphene device, and basically they have some kind of radio frequency tag, and they can sense some kind of molecules directly on top of graphene. So that would be an interesting approach, but we haven't studied that yet. Thanks for the suggestion.
Great.
Okay, so are there any additional questions for Juan? So I guess we're still 10 minutes early.
Okay.
And that's a first for me in a conference, to be early during the first few talks. But anyway.
Well, what is the current status, Juan?
What is that?
What is the current status of your device? Actually,
yeah, we were planning on making some measurements in the lab. Like we were trying to get, actually during this pandemic time, we got a machine for producing graphene oxide.
Okay.
That was going to be an approach that we were going to use for measuring volatile gases. That was from another project. But we were also interested in seeing if we could actually functionalize this kind of surfaces and see if we can measure.
Yeah, I'm worried about the facilities do you need to implement this device. I think it's kind of hard. I think it's because it's not a crystal, it's not amorphous. This is a chemical process, I think. And then what kind of lab do you need for this kind of?
No, we can.
Not really.
We've been producing graphene for a while already. You can reduce graphene from graphene oxide, so that you can place in liquid form on a plastic, and then you evaporate the liquid. You're left with a graphene oxide, and then you can use a laser at 780 nanometer wavelength to remove the oxide, and you're left with graphene chips that can be used again as a 2D material for sensing. So that's straightforward, right, Juan?
Yes, and one of the ideas of this design is that we can, we try to use minimal equipment and minimum cost as possible. So that's why, we're trying to explore all these possibilities where we can. Of course, we could just buy commercial devices, but those are $400, $500 per chip. So it's probably not the best solution, but we are actually trying to use one of those just as a proof of concept. And because as I was mentioning in the talk, it's a lot easier if we can have metallic graphene. So one of the ideas is try to use this device and see later we will try to find a more cost-effective solution for that.
But then based on your responses, then it caused me another worry, because how sensitive is this device to very little changes in just one single atom or one single layer, maybe change the complete design? Then how sensitive is for maybe this kind of application?
From calculations in a single device, we get like difference in a couple of maybe 1 microamp between one molecule and the other. So it would be a challenge to measure, but I don't think it's impossible. It would be more difficult if the difference was less. But we will also try to continue working on the electronics. So hopefully Sammy will give us an idea of the talk later on.
Great work.
I think one of the things you didn't show, Juan, is the fact that using the ligands, you're reducing the thermal noise in the system, which was an initial worry for these. And by reducing it, you were able to calculate how much deviation in terms of the height of the molecule with respect to the channel would affect your actual electronic signature. And you were able to show in the paper that it was actually within the standard deviation of the discrimination signal between 1, 2, and 3 UDP glucose molecules on the channel. So again, I think the idea for these devices is not to make a single device, but to make an array of these devices that could be used as basically on-off switches to distinguish the presence of a molecule in each bit of that array.
Okay.
But
great work.
Yeah, good work.
Great work. Congrats.
Okay, so I think we're still early, but I think we're gonna move over to the next talk if we don't have any other questions. Then we will take a break, and probably a longer break than the usual. But let's move on to the next talk. Thank you, Juan, again. Our next talk is by Sami Perdomo. Sami is an electronic engineer. He's currently pursuing his PhD, and in basically in electronics and biosciences. He's combining both to look at biosensing. And Sami is going to talk today about his development of a potentiostat, which is an instrument required to read electrochemical devices. And that's when you have biomarkers in solution and you need to actually solve the measurement of electrons as they cross an interface between the solution and your electrode. So Sami will be discussing the current status of his development. Sami, please go ahead.
Muchas gracias a todos por asistir a esta charla en la cual voy a mostrar algunos resultados y avances que he tenido dentro del proyecto que estoy desarrollando como parte del programa de Omics y parte de mi aprendizaje como estudiante de doctorado de la Universidad Javeriana. Así que pues vamos a empezar. En primer lugar, pues voy a hacer un pequeño repaso o mencionar algunos conceptos importantes para que las personas que no están relacionadas con el tema pues puedan entender cuál es el objetivo que pretendemos desarrollar con ese proyecto. Voy a definir como tal qué es un nanosensor, que un nanosensor es un dispositivo que se basa en tecnología a nanoscala para detectar o para medir muy bajas concentraciones de algún tipo de analito, y cuando digo analito quiero decir algún tipo de molécula que es de mi interés determinar, ¿sí? Para poder expresar en una cantidad cuantificada qué valor o qué concentración hay en un determinado volumen o en un determinado espacio, en un determinado sistema. Así que generalmente con un nanosensor podemos llegar a determinar o a medir concentraciones que están por debajo de los nanomol por litro, ¿sí? O sea, es algo extremadamente pequeño. Y poder entender esto y poder determinar estas concentraciones permiten, por ejemplo, que se pueda entender lo que son los distintos procesos biológicos a una escala que antes no era posible elucidar o entender. Así que generalmente este gráfico que tengo acá representa lo que es un nanosensor. Él se compone de tres elementos básicos que son el electrodo, el transductor y el detector. Eh, en el electrodo es donde va a ocurrir todo el proceso químico y físico en el que una molécula llamada ligando, ¿sí? Puede asociarse o se va a asociar al analito que yo quiero encontrar, dado que son químicamente muy afines entre ellos mismos. Entonces, cada vez que ese ligando detecte ese analito, esa molécula que yo quiero detectar, el electrodo va a emitir una respuesta o una información que va a ir al transductor. El transductor lo que me va a hacer es convertir esta información a otro tipo de información medible, que esa va a depender de la tecnología que nosotros estemos utilizando. Si es una tecnología de sensores ópticos o electroquímicos o de sensores de transistor o sensores mecánicos, ¿sí? Cada uno tiene un principio de operación distinto. Y esa señal del transductor va a llegar a un detector que es mi sistema electrónico, el cual va a tomar la señal y la va a interpretar para darme una cantidad medible y cuantificada, ¿sí? Equivalente a ¿qué? A esa concentración del analito que yo estoy buscando. Bien, ahora voy a mencionar un breve resumen acerca de las tecnologías más importantes que son de los nanosensores y voy a empezar por lo que es los nanosensores ópticos. Entonces, dentro de los nanosensores ópticos vamos a encontrar una interacción, ¿cierto?, entre los analitos y un haz de luz y una luz. Entonces vamos a tener dos tecnologías pues, que resalté acá como para explicar su funcionamiento, que son las tecnologías de SPR o Surface Plasmon Resonance, en las cuales el principio se basa en que yo voy a tener una capa o una lámina de metal sobre la cual voy a funcionalizar, ¿sí?, la superficie para asociar el analito y los ligandos o el complejo ligando analito que yo quiero evaluar. Y lo que va a suceder es que dado que en la superficie de una lámina metal hay bastantes electrones libres, cuando un haz de luz incida por el otro extremo del metal, va a hacer que empiecen a presentarse oscilaciones de estos electrones que son conocidas como los electrones plasmones o plasmons, ¿sí? Y eso va a causar que en dependencia de la cantidad de analitos que hayan asociados a los ligandos de la superficie, el ángulo acá reflejado, ¿cierto?, que yo obtengo del haz de luz va a verse desplazado, ¿sí?, en el espectro, es decir, va a haber un corrimiento en dependencia de esa concentración que yo tenga acá. Entonces, de esta forma yo puedo determinar qué concentración de analitos tengo asociados en esta parte. Ahora, tenemos otro tipo de tecnología que es la tecnología por fluorescencia, en la que yo voy a utilizar materiales que tienen una propiedad óptica fluorescente, es decir, que emiten luz en presencia de una excitación de energía, ¿sí?, en este caso de un haz de luz, y voy a ver que se incluyen y se utilizan lo que son nanopartículas, ¿sí?, por lo general nanopartículas de oro, las cuales lo que sucede es que por fluorescencia yo voy a asociar a mi nanopartícula unos fluoróforos que van a responder a un estímulo de luz. Entonces, cuando está actuando acá las de luz, ¿cierto? El fluoróforo emite luz, pero ocurre un efecto de ensombrecimiento causado porque la nanopartícula de oro absorbe esa radiación. Entonces, en conclusión, no voy a encontrar que haya luz reflectada a partir de este sistema. Pero cuando llega y se me asocia el analito que yo quiero, va a desplazar la nanopartícula, va a aumentar la distancia entre la nanopartícula y el sistema, por lo cual ya ese efecto de ensombrecimiento o quenching, ¿sí?, va a verse disminuido y va a empezar ya no a absorber la luz que estaba reflejando, sino que irradiándose, sino que ahora ella va a aparecer y voy a detectar una intensidad de luz equivalente a la concentración de moléculas o de analitos que yo tenga asociados a los ligandos. ¿Listo? Tengo por otro lado lo que es los nanosensores trabajados en lo que es la tecnología de los transistores FET, en los que básicamente yo voy a tener un canal, ¿cierto? Que va a ser de un material que va a tener propiedades electrónicas que van a depender de ¿qué? De dos cosas, de estos electrodos que yo tengo de drain y source, los cuales van a polarizar en el sistema y lo que van a hacer es generar una densidad de estados a través de las cuales a través de este canal pueda fluir o existir un flujo de electrones o una corriente. Entonces, en dependencia de la cantidad de analitos que yo tenga asociados en la superficie del canal, esas propiedades eléctricas de ese canal van a cambiar, desde su conductividad van a cambiar, y por ende yo puedo regular por este electrodo de gate, ¿sí? La cantidad de corriente que yo pueda permitir salir a través del canal. Entonces, de ese modo yo puedo detectar a través de un cambio de corriente lo que es la concentración o la asociación de los analitos con los ligandos. ¿Listo? Y tenemos los sensores electroquímicos que ellos se basan prácticamente en una celda electroquímica, ¿cierto? Donde voy a tener generalmente un arreglo de 3 electrodos, los cuales son el electrodo auxiliar, el electrodo y el electrodo de trabajo. Sobre el electrodo de trabajo es que vamos a generar la asociación del ligando y del analito de interés, y lo que se va a medir en realidad es la dinámica de las reacciones de oxidación y reducción que se presentan acá en este complejo. Estas reacciones las podemos medir a través de las distintas técnicas electroquímicas, como son la potenciometría, la espectroscopía de impedancia o la voltametría. Entonces voy a tener un sistema que va a ser un potenciostato, que es el que me va a permitir trabajar como el detector que mencionamos anteriormente, y él obtiene la información que viene acá de este sistema electroquímico y lo convierte a una cantidad medible equivalente a lo que es la concentración que buscamos. Hablando del potenciostato, pues generalmente un potenciostato tiene que cumplir dos funciones principales, ¿sí? La primera función es asegurar un voltaje, ¿sí? Constante sobre la celda electroquímica, ¿sí? Por lo cual yo tengo mi voltaje de entrada, que es el que voy a aplicar y depende de la técnica electroquímica. Voy a tener voltaje de referencia, que si nos damos cuenta está acá en un seguidor. Entonces ese seguidor va a generar un potencial constante en la celda. ¿Listo? Y la otra tarea que tiene que realizar este potenciostato es permitir la medida de la corriente que se genera o que se transmite a partir desde el electrodo auxiliar al electrodo de trabajo, ¿sí? Y esa corriente va a ser la que yo tomo de información para obtener los resultados deseados dependiendo de la técnica que yo esté evaluando. Actualmente existen pues distintos potenciostatos, ¿sí? Desde unos que son robustos a unos que son portables, pero una de las desventajas de estos equipos es que son altamente costosos. ¿Listo? Eh, Ahora, ¿cuál es el problema? El problema es poder realmente buscar, ¿sí? Un modo en el que se pueda generar una plataforma que me permita a mí integrar las tres tecnologías principales de nanosensores y generar a través de ellas un sistema de medición sin importar el analito que esté evaluando. Es decir, si yo tengo un analito X y quiero evaluarlo y quiero medir cómo es el desempeño de medida, tanto en la tecnología utilizando transistores FET como utilizando ópticos, como utilizando electroquímicos, yo puedo tener una plataforma y esa plataforma caracterizarse por ser de bajo costo y altamente portable para evaluar en mis tres tecnologías cuál es la mejor respuesta que obtengo para medir y determinar ese tipo de analito que estoy evaluando. Entonces, en realidad, el problema está abarcado es cómo yo puedo integrar estas tres tecnologías en un solo dispositivo, ¿sí? De medida. Ahora bien, hablando y mencionando algunas cosas del estado de arte que se han revisado, realmente ya han existido trabajos en los que se han desarrollado sistemas de medida, ¿sí? ¿A partir de qué? De la tecnología que se haya usado de nanosensor. Por ejemplo, en este trabajo de Lee, lo que se utilizó es utilizar un sistema de medida, ¿sí? De acondicionamiento electrónico para tomar la señal de este transistor, ¿sí? La señal de corriente, y llevarla a un sistema que me permitiera visualizarlo en un computador o en un osciloscopio. Vamos a explorar otro trabajo en el que una combinación, ¿cierto? Ahí ya comenzamos a ver las ideas de integrar no solo una tecnología, sino varias tecnologías de nanosensores, y en este caso, en el trabajo de Yu, lo que se hizo es integrar la tecnología de los FET con la tecnología de los sensores ópticos para generar un sistema de medición de pH a partir de un transistor FET y de un sistema óptico. Entonces lo que se hizo es adicionar sobre el canal del FET fluoróforos que eran excitados a través de este LED, y esa respuesta cuando se cambiaba la concentración de pH era adquirida a través de un sistema de lentes, ¿cierto? Y una fibra óptica para ser convertido a una señal eléctrica equivalente y así poder comparar ambas respuestas, ¿listo? Entonces lo que se hizo acá, digamos, volvió y se remarcó el cambio de la corriente que yo estoy midiendo a un potencial equivalente, solo que en este caso se utilizó una estrategia del puente de Wheatstone. Lo que se muestra en estas gráficas es que la sensitividad para la técnica de fluorescencia y la técnica del FET son prácticamente iguales. Y yo cuando uno estos dos tipos de información, obtengo una curva de sensitividad mejorada en comparación con cada una de las dos individuales. El siguiente trabajo de Lee et al. De lo que ellos hicieron fue nuevamente también un sistema similar, solo que en este caso integraron lo que es la tecnología del nanosensor electroquímico que tenemos acá con la tecnología de los sensores FET. Entonces, el sistema de ellos era controlado por una tarjeta FPGA que conmutaba cuál sistema de medida se iba a utilizar. Lo importante de este trabajo es que nuevamente existe esa conversión de corriente a voltaje, pero en este caso con una topología utilizando un amplificador operacional. Entonces esta topología se conoce como un amplificador de transimpedancia, ¿sí? Y lo que hace él es convertir la corriente que pasa a través de esta resistencia, ¿sí? En un voltaje siguiendo la ley de Ohm. Bien, entonces para desarrollar la metodología evaluada, lo que se realizó, o para desarrollar este sistema, ¿sí? Lo que se empezó primero fue por analizar todas esas condiciones de ruido, empezando desde qué, desde evaluando cuál era realmente el sistema equivalente o el circuito equivalente de cada una de las tecnologías. Representar mi transistor FET como un arreglo de una resistencia para una capacitancia y siendo la resistencia de mi canal. ¿Sí? La capacitancia puede estar relacionada con la capacitancia cuántica o el dieléctrico que se produce ahí, y voy a ver que lo que obtengo yo es al final una variación de corriente. En mi sistema electroquímico lo mismo, en mi solución tengo una resistencia electrolítica, voy a tener unas capacitancias que van a darse por la interfaz entre la solución y el electrodo, que es la capacitancia de doble capa que se conoce, y voy a tener unas resistencias de difusión. Entonces, al final lo que voy a obtener es también una variación de corriente. Y en mi sistema óptico yo lo puedo representar a través de ¿qué? De un receptor óptico, ¿sí? O un diodo de recepción de luz, que lo que va a hacer es generar una variación de corriente, ¿cierto? En función de la intensidad recibida. Entonces, al final podemos observar que la corriente es una variable común para todas las tecnologías. Por lo tanto, podemos pensar en aprovechar la medida de esa corriente para integrar los tres sistemas. Ahora, este es el diagrama de bloques que se propuso para el sistema, ¿sí? En el cual vamos a tener como procesador central una Raspberry Pi que nos ofrece distintas ventajas como su capacidad de procesamiento, la memoria RAM que tiene, y tenemos también una facilidad de generar una interfaz de usuario a través de ella. Incluso integrar lo que es un LCD táctil de 2.8 pulgadas, que es donde el usuario verificará e ingresará los datos y verificará los resultados, y vamos a tener tres módulos distintos. El primer módulo es el módulo electroquímico, ¿cierto? El segundo módulo será el módulo de control del FET y el tercer módulo será el módulo de control del sensor óptico. El módulo electroquímico necesitará de la existencia de un generador de señal, que es el que me va a generar la señal de la técnica electroquímica que voy a aplicar, ¿listo? Y va a tener un circuito de front-end para recibir ¿qué? La señal de corriente de los electrodos. Recordemos que estas señales de corriente son de baja intensidad, casi estamos abarcando desde el rango de nanoamperios a microamperios, por lo cual tiene que tener un circuito de acondicionamiento lo suficientemente robusto para poder permitir una buena medida. El sistema FET vamos a tener un control DC que se realizará a través de un DAC, y ese DAC va a generar los potenciales para los potencial de los electrodos, tanto de gate como de drain, y vamos a tener un sistema óptico que lo que va a evaluarse y, bueno, lo que va a recibirse es la respuesta del sistema cuando se haya excitado a través de una luz LED, ¿cierto? Trabajando aproximadamente en el rango de 400 nanómetros, y la respuesta del receptor es la que vamos a transformar, y ese receptor óptico trabajará en el mismo rango. Bien, a partir de ciertas especificaciones, lo que se realizó es empezar a desarrollar el módulo electroquímico. Entonces, este módulo electroquímico, lo que se comenzó es en reunir la información de distintos potenciostatos comerciales, ¿listo? Y sacar un estimado de especificaciones que nos permitían iniciar el diseño. Entonces, estas son especificaciones a partir de las cuales nosotros basamos el diseño, por ejemplo, los rangos de potenciales DC, los rangos de frecuencia, todos estos sirvieron como base para desarrollar, por ejemplo, el sistema de generación de señal, y también, por ejemplo, la frecuencia de muestreo para la captura de los datos, ¿listo? Y los rangos de corriente para ese rango, ese control de corriente que mencionamos anteriormente. Las dimensiones del sistema están basadas en el tamaño de los componentes más grandes. En este caso sería el LCD de 2.8 pulgadas y la Raspberry Pi, que serían 90 milímetros por 60.5 milímetros por 50 milímetros, que es un sistema pequeño, ¿sí? Y que esto obviamente nos puede dar nuestra portabilidad, ¿sí? La portabilidad también definida por la batería que estamos utilizando, que nos da aproximadamente unas 6 horas de uso continuo aproximado. Bien, entonces el siguiente paso fue desarrollar el sistema de generación de señal. Para eso partimos a partir de un módulo de generación de señal que fue el integrado 2206, que es uno de los integrados, digamos, más comunes, pero que son de bajo costo, y lo que se hace es configurarlo de tal modo que la frecuencia viene determinada por la relación de unos RC, y RC está determinado a un pin dedicado, ¿sí? Es decir, yo puedo controlar la frecuencia con esa resistencia y con la capacitancia dada entre estos pines, ¿listo? Entre el pin 5 y el pin 6. Entonces, a partir de eso, pues se diseñó un sistema de variación de frecuencia, de tal modo que se calcularon las capacitancias requeridas para trabajar dentro de los rangos de frecuencia deseados, ¿sí? Y lo que se hace es realizar una red de conmutación de tal modo que a través de la Raspberry Pi se decía en qué rango de frecuencia se quiere trabajar. Por ejemplo, quiero trabajar de 1 a 10 Hz, voy a activar el capacitor correspondiente, y lo que voy a hacer con la resistencia es una resistencia que va a estar controlada digitalmente, es decir, un potenciómetro digital que a través de la Raspberry, ¿sí? Puedo controlar el valor de la resistencia que yo deseo, y con eso ajustar el rango de frecuencia y moverme dentro de ese rango de frecuencia a partir de la variación de esta resistencia. Del mismo modo, la variación en amplitud de la señal se da a través del pin 3. Esta amplitud de la señal es a través de valores resistivos, por lo cual se utiliza simplemente un potenciómetro digital para generar esa variación de amplitud controlada desde la Raspberry. ¿Listo? Y lo que observamos acá es ya una prueba de 100 a 1 kHz, es decir, un barrido de frecuencia que se realizó para observar cómo era el desempeño de la onda senoidal. Y acá vemos la señal triangular que se genera, que es posible controlarla a través de la amplitud que mencionamos, y la cantidad de muestras que adquirimos nos permite controlar el número de ciclos o scans que yo puedo tener. Listo, entonces vamos a hablar ya de ese amplificador de transimpedancia que lo vimos que es fundamental en la literatura, que es el que nos va a permitir transformar la señal de corriente que yo mida de mi sensor, sin importar cuál sea, y transformarla en una señal de corriente equivalente. Entonces lo que se hace es que, dado que la impedancia a la entrada del amplificador operacional es tan grande, la corriente prácticamente pasa a través de la resistencia, ¿sí? Y siguiendo la ley de Ohm me da un voltaje equivalente a esa corriente, ¿listo? La resistencia es un valor constante. Lo que hace el capacitor acá es disminuir los efectos de ruido térmico. Un componente resistivo me aporta al sistema, y esto nos permite entonces tener una fácil conversión de una corriente a un voltaje, que es el que yo voy finalmente a adquirir. Para la espectroscopía de impedancia, lo que se adicionó fue una etapa, de esta etapa que tenemos acá, que es una etapa, pues, es un sistema, un circuito de integración. Lo que hace es dar una amplificación o una preamplificación sin componentes resistivos, dado que la señal de corriente en las técnicas dado que el potencial que se utiliza en las técnicas de espectroscopía es muy pequeño, entonces lo que va a hacer es amplificarlo, ¿cierto? Con estos con la relación de estos dos capacitores en la enlace de realimentación y a la salida del primer amplificador operacional. Y esa amplificación se me va a dar, ¿qué? O sea, un control de corriente sin incluir lo que es ruido térmico, ¿sí? Es puramente capacitivo. Después de eso de amplificar, si lo llevo a mi sistema de amplificación de transimpedancia, donde entonces convierto la corriente que estoy midiendo, acá estoy haciendo en la primera etapa una conversión, amplificación de corriente a corriente, y en la siguiente etapa hago la conversión de corriente, esa corriente que amplifique, ahí sí a un voltaje que pueda medir, ¿listo? Entonces, ya cuando tenemos el sistema de procesamiento, adquirimos la señal, lo que hacemos es un procesamiento digital para poder encontrar qué, ajustar la señal que tenemos en el fondo, la señal que recibimos, ¿cierto? Del electrodo, es decir, de la espectroscopía de impedancia, y lo que hacemos es ajustarla a una curva que tenemos. Hacemos un ajuste de la curva, ¿sí? Para este caso utilizamos un algoritmo de gradiente descendente en donde determinamos las amplitudes a través de la identificación de picos, ¿sí? Y la única variable para el ajuste de gradiente descendente es la fase. Entonces, midiendo el error cuadrático entre una señal base, una señal sin fase, y la señal deseada, entonces lo que hacemos es hacer el algoritmo de aprendizaje para encontrar el parámetro óptimo de fase. Esa fase pues se utiliza para poder determinar el desfase entre la entrada y la salida, ¿sí? Recuerden que vamos a medir una impedancia, la impedancia es la relación entre el voltaje de entrada y la corriente de salida, y la fase que hay entre ellas dos. Entonces, a partir de eso, pues el algoritmo me calcula la amplitud de corriente, la amplitud de voltaje, la magnitud de impedancia, la fase, ¿sí? Y los componentes real e imaginarios de esa impedancia. Entonces, lo que vamos a mostrar acá es unos resultados preliminares que tenemos, ¿listo? Ya se han realizado algunas pruebas, se realizaron pruebas con ferricianuro, ¿sí? Como muestra, y con electrodos serigrafiados, los cuales pues me dan la facilidad de conexión, ¿sí? Ya tenemos ahí tanto el electrodo de referencia como el electrodo de trabajo, como el electrodo auxiliar, ¿listo? Y estos son los parámetros que utilizamos, el voltaje, el offset, la frecuencia que se utilizó, y el scan rate de la señal triangular y el número de scan. Y esta fue la respuesta del sistema. También se hicieron pruebas de la espectroscopía de impedancia, lo mismo, se utilizó como muestra el ferricianuro, se utilizó entonces el mismo tipo de electrodo, el screen printed electrode, y estos son los parámetros que se utilizaron. El barrido se realizó de 0.9 Hz hasta 10 kHz y obtenemos el espectro que vemos a continuación. Entonces, lo mismo, se realizó una comparación entre el espectro obtenido. Esta primera gráfica es la componente real, la segunda es la componente imaginaria. Los resultados de la izquierda son del componente que se desarrolló y los de la derecha son del componente de un equipo comercial que es el PAN-SENS. Esta es la final, es el diagrama, el espectro de impedancia completo, que vemos que tiene una dinámica similar, ¿listo? Y lo que nos demuestra es que realmente se puede obtener una buena aproximación a partir de este diseño con un equipo de bajo costo portable en comparación con un equipo robusto, por decirlo así, y que es pues comercialmente más costoso. Listo, entonces hasta aquí pues es los avances que hemos logrado. Lo que sigue ahora es seguir trabajando en las partes de los otros módulos de sensado, mejorar algunos aspectos del módulo electroquímico que llevamos hasta ahora, pero la idea es continuar con el trabajo. Entonces, muchas gracias a todos por su atención. Si tienen alguna duda, pues quedo atento a sus preguntas. Muchas gracias y hasta luego.
Well, Thank you very much, Sammy, for your talk. Very interesting. We are now open for questions in our chat. I will start as we don't have any yet. So, Sammy, the whole point of doing fusion, sensor fusion is really to be able to capture different signals at in different universes of discourse. So you would have an optical, let's say a fluorescence-based device, contrasting signals with a device that gives you electronic transport or gives you redox information at the solute-solvent interface. How do you think that's going to play in the end if all three schemes or techniques have very different limits of detection? Do you think they can complement, supplement each other? What do you think will happen?
Yes, so the idea is integrate all these technologies to find a point in which we can really compare how they improve the detection of the analyte of interest. So we can, I don't know, if I find that the FED technology complements the optic technology. So this will be possible if I really can measure and in real time and the same time the two technologies with the analyte. So this is the only way that we can really say if that technology can complement other technology. So in my opinion, I can hope that really the technologies can complement how we how we find in the state of the art. So many researchers are at this moment integrate these technologies to improve the debit of detection that can be get with any technology individually.
Okay, and what do you see as the main drawbacks of each technology or main problems, and how having a complementary technology might be able to cover those drawbacks initially? So let's say you have a field effect device with the option to do optical fluorescence and with the option to do electrochemistry. How do you think the limits of electrochemistry would be able to would be covered by the field effect device or by the fluorescence, and so forth? How would the limits of the fluorescence-based system be complemented by the other two? Because that's the main point. Or you could also have additive information. You basically add the signals. You have to normalize them in some in some unit, and or transform them and then normalize them to be able to add them and obtain from the from the added signals some information that is not present in the individual signals. How do you think that's going to play out?
Yes, how I mentioned in the state of the art, one of the articles find that summarizes the sensitivity of the two technologies that they integrate. They get a better or are improved sensitivity based on the technologies used. So I think that, for example, each technology measures different dynamics of the molecule that is interacting with the proof. So we can complement that dynamics that is measured for each one. So for example, in the electrochemical technique, I measure a different dynamics that is the, I don't know, for example, the diffusive part of the association with the analytic of interest. In the FED, I can get a different answer about these dynamics, because in that technology, I measure the interaction with the ligand, and this interaction will change my conductivity with the channel. So I think that, for example, each technology has different kinds of of measure or measurement time. So this time can be complementing each other depending on the dynamics of the system.
Okay. And what's going to be your first analyte? What are you going to test as your first analyte?
The first analysis that we will test with this device is the COVID antibody protein in the Sensum SARS project. Yes.
Okay, great. Sami, thank you very much. Let's see if we have any other questions. Yes, there's a question by Victor Rodriguez Toro. Thanks for the presentation. Are you planning to measure the noise of your sensors, or do you plan to use models for quantifying the noise?
Yes, actually, we have models that we found in the state of the art that model the noise based on the transimpedance amplifier. So this was the first step, really measure through a mathematical model what will be the noise in the system. And this model really is really, okay, the signal to noise ratio that we get with this system is really low, because how I mentioned in the presentation, when we use an operational amplifier, we can take these parameters of noise and be manipulated so easily, because the properties that that have these devices, for example, the low input impedance of or the high impedance at the output. So yes, we have models that model that noise that you are asking for.
Okay, Sami, thank you. Any other questions from the audience? If not, let's thank Sami once again for a good presentation.
All of you.
Okay, and we're going to be moving on to our next presenter, our next talk. Our next talk is by Gustavo Lara. Gustavo is currently pursuing his doctoral degree, and he's focused on a particular problem that is very challenging for small molecular weight analytes, and that is trying to be selective in detecting the target analyte, and how to improve selectivity and sensitivity to that particular analyte. So he's going to be talking about his progress in sucrose detection and low concentration sucrose detection in plants and in tissue. So Gustavo, we're all yours. Thank you.
I will present part of my work as a graduate student here in Javeriana, Cali. My work is related with the in silico design of ligands for the detection of sucrose at ultra lower concentrations in physiological conditions. And this work is under the direction of Professor Andrés Jaramillo. This is my presentation agenda. And as I mentioned, we are interested in the detection of sucrose. This molecule is formed by one molecule of glucose, and the other molecule is fructose. And this is one of the principal mechanisms for the fixation of carbon in the trophic chain. This molecule is a very stable molecule because this is a non-reducing sugar, and probably this high chemical stability is the main reason why this molecule plays several key roles in plants, as, for example, regulator for the growth and development, and also is used as precursor for the synthesis of new molecules in non-photosynthetic tissues. And for us, apart for the importance of this molecule in nature and for its importance in plants, this molecule is important for us because this is the principal commodity of our agro-industrial complex. Then the biosynthesis of sucrose begins in plant leaves, inside the chloroplast. In the chloroplast, when the Calvin cycle where the Calvin cycle produce triose phosphate molecules, and these triphosphate molecules pass from the chloroplast to the cytosol, and in the cytosol, several reactions produce as main product the sucrose molecule. Once this sucrose is synthesized, it is transported from the leaves to other non-photosynthetic tissues, and in these tissues this sucrose is used to produce cellulose, or is used to produce starch, or is used directly as as one form of to storage to save energy, or is used in the mitochondria to produce energy. This is an scheme how this sucrose can be decomposed again in glucose and fructose to form, for example, trehalose. Trehalose is an important feed hormone in plants that regulates a lot of process, or this sucrose can be used to produce again ATP glucose, use it to produce cell-wall polysaccharides, or after several reactions, this ATP glucose can be used to produce starch or maltose. Then there are a very complex relationship between glucose, sucrose, and other sugars in plants. And these sugars sometimes have important roles in the plants. For example, concentrations of glucose around 25 millimolar reduce the starch synthesis in germinating seeds, or the concentrations about 25 millimolar of trehalose inhibits growth elongation in Italiana seedlings. Also, it has been reported that genes that encode enzymes in the metabolism of sucrose are also involved in the production of sugar signaling, and these sugars also can be produced by sucrose. Then we have here a very complex feedback system for the regulation of many genes and expressions in plants, and also that are part also for the response of these plants to some kind of stresses. Then the detection of some of the sugars in different plant tissues is a key task to understand these complex relationships, and the detection of different analytes inside the cell. It could be a complex task because we have a lot of different molecular entities, a great variety. For example, in bacterial cells, the number of different sugars and precursors are about 250. Probably in plant cells, this number is even bigger. Also, the characteristics of the system, of the matrix for the detection of the analyte means that we should be able to identify the hydroxyl groups in the carbohydrates and differentiate these hydroxyl groups from the water molecules, because inside the cell is a system that is highly solvated. Usually this is archived with the usual techniques of sensing throughout the sample pre-treatment and processing, but this means that the tissue should be destroyed to separate the analyte of interest from other substances that interferes with the measurement. And one way to circumvent these problems is the in vivo detection of the analyte. And for this task, we should have sensors that met these characteristics. For example, the response from the sensor due the detection of the analyte should be a reversible response. And this is related with the with the balance of the homeostatic conditions. If we took all the analyte for the measurement or for the detection with the sensor, probably we will lead the system to an homeostatic imbalance. Also, these sensors should work in physiological conditions. And as I showed you, this sensor should have a high sensitivity and selectivity due to the great variety of molecular entities inside a cell. For the detection of in vivo for the detection of in vivo for the in vivo detection of sugars, some systems have been developed. For example, lectins. Lectins are proteins that have the advantage of high biocompatibility, but some difficulties are related with the selection of the template to get a high selectivity to different polysaccharides. These lectins interacts with the carbohydrates throughout non-covalent interactions. Also, aptamers have been developed to the sensing of sugars. These systems also interacts with sugars throughout non-covalent interactions. Aptamers are systems with less than 100 oligonucleotide bases. And also these systems have the advantage of high biocompatibility, but in the case of aptamers, it has been seen that its performance is improved when the sugars has a net charge, that because this improves its interaction with the aromatic rings in aptamer. But for this condition can be filled by simple sugars as sucrose. And other of the systems developed to the detection of in vivo conditions for sugars have been the use of phenylboronic acid. The systems interacts with the oil groups in the sugars, and these groups interacts or form covalent bonds with the hydroxyl groups in the 1,2 and 1,3 relative positions. This system has the advantage of versatility for integration into different platforms of different sensing platforms as polymers and nanotube and aptamers. But one of its disadvantages is that its binding energy is improved when the pH concentration is far from the homeostatic conditions. But taking into account these pros and cons for these systems that have been studied for the in vivo detection of sucrose, we have decided to use the phenylboronic acid system because we think that it is possible to improve its performance if we are able to control precisely the orientation of these phenylboronic groups to the selectivity bonding of the hydroxyl groups in sucrose. Also, we think that that this system forms reacts reversibly reactions with sugars, then this will help with the balance, homeostatic balance during the detection of the analyte. Then we have characterized this system from the reports of the literature. It has been reported that in aqueous system, the phenylboronic acid has its most stable form when the boron atom is in sp3 hybridization. Also, it has been reported that the stability of the molecules or the molecules or the complexes between the phenylboronic acid with hydroxyl groups, the hydroxyl groups improves when the boron atom is bonded to functional groups that are able to stabilize the negative charge when the boron atom changes from sp3 to from sp2 to sp3 hybridization. We also have characterized the physical chemical properties for the sucrose. Sucrose has been reported that sucrose has 4 principal conformers, and in this report, they have calculated the relative energy between these conformers, and they have found that the most stable conformer is related. They name it S1 configuration, and these results are consistent throughout different levels of theory for these calculations. Here, these authors report some classical simulations for glucose, where they calculate the free energy landscape. This point here belongs to the configuration of sucrose in the S1 conform. Here they are reporting the results for the calculation of dipolar residual coupling during its trajectories. The points here in green means that they got a very good correspondence from the dipolar residual coupling calculated with its trajectory and reported directly from experiments with NMR. And here the authors are also reporting the distribution of the hydrogen bonds in sucrose with several levels of with several explicit solvents. After this literature review, now we have a scaffold to design our sensor or sucrose detection with phenylboronic acids. We use the S1 configuration, and to this configuration, this conformer of sucrose, I have calculated the binding energies of these boronic acids to the different groups to the different hydroxyl groups in sucrose. Here I am reporting these binding energies for two phenylboronic groups bonded to the sucrose. In blue, I have labeled the hydroxyl groups in the glucose moiety, and in red, I have labeled the hydroxyl groups in the fructose moiety. Then we have found that for these configurations, here the R and S configurations means the stereochemical configuration of the boron atom. This is a chiral atom when the boron when the boronate complex is formed. The boronate complex is the boron attached or bonded to the sucrose molecule. Then we have favorable or exothermic enthalpy reactions for the union of phenylboronic groups, of two phenylboronic groups to, for example, to the 1,3 hydroxyl groups in fructose and the 4,6 hydroxyl groups in glucose. This with the R and S configuration. And in this case, we have a enthalpy, a reaction enthalpy of minus 9.8 kilocalories mole. We have also calculated these bonding energies in these hydroxyl groups using the benzopyroxole. In this case, these binding energies are are around 4 kilocalories more. And in these cases, for the sucrose, we don't have find any exothermic reaction for the union of the phenylboronic groups to relative hydroxyl groups in positions 1, 2, and this is related with the fact that these hydroxyl groups are in trans positions, and when the phenylboronic acid is bonded to these hydroxyl groups, then we will have a half chair configuration for these five atom rings. And this is an unstable configuration compared with this configuration that is called envelope. But for the case of interactions, 1,3 interactions between phenylboronic acid with the hydroxyl groups in sucrose, we have found that for the bonding with the hydroxyl groups 4 and 6 in sucrose, in this case, the 6-member atoms ring form has a chair configuration, the most stable. But for example, in the case of 4,6 hydroxyl groups in sucrose, I have found that in that case, it's not possible to get the chair configuration, and instead a twisted boat configuration it is formed. Here and then, this is one of the molecules. One of the molecules where the reaction enthalpy is exothermic. Here we can see the chair conformation for these 6 atom rings. Also in this case, we can see here a chair conformation for this 6 atom ring. And once these sites for the union for these sites favorable to the union of the phenylboronic acids was found, then the next step was the calculation of some backbone to bridge both moieties, both phenylboronic acids, to form now the complete Wittenstein ligand conform just for carbon atoms. For this DAC, I use the overlay program inside the Health Designer suite. I should for this in this program, I should choose some hydrogen bonds that will be used for the formation of or the calculation of the backbone of carbon atoms. For this case, then I choose these hydrogen bonds enclosed by this red circle. And this program follows some some rules to form this bridge between these two entities, or between the two moieties in the of the phenylboronic groups, of both moieties of phenylboronic groups. Sorry. Then we have found that for this configuration of the phenylboronic groups attached to the hydroxyl groups 1,3 in fructose, and the glucose, and the hydroxyl groups 4,6 in glucose, we have found just one ligand structure that bridge these both phenylboronic groups, and the system has a binding energy of minus 9.8 kilocalories mole. For this configuration, we have found around 467 ligand different structures with energies. This energy is the energy that we have calculated for the reaction enthalpy just with the phenylboronic groups. This Rundin square deviation means the difference between the bond, the difference between this bond when we have just the hydrogen atom, and when the carbon backbone to attach these molecules was calculated. For this case, we got 7 ligand different structures, and in this case we got 2 ligand structures. I am just reporting the structures or the ligands for which I have I got some bridge for the union of the both moieties. In the case of benzoperoxole, we didn't got any structure. Then, as a result, we have designed ligands for the specific detection of sucrose under physiological conditions, and we thought these systems have the potential for its application in vivo conditions. The ligands designed has binding energies around 8 kilocalories mole, similar to some lectins and polysaccharides, and I thought this binding energies at the starting point to improve the system is a very good starting point. And we thought that the system or the binding energy can be improved if we add some chemical species that are able to bond with the free hydroxyl groups in sucrose through Gauss covalent interactions, and also the addition of electron drawing groups to the benzoberol could improve the binding energy, but care should be taken to get a system that don't lead the system, the cell or the cell to an homeostatic imbalance. I want thanks to our sponsor by funding this project, the Alliance, the Omics program, and also want to thank to all of you for your time and for your attention.
Well, thank you, Gustavo, for a great talk. In the meantime, while we receive any other questions, I have a few. So it's a very interesting approach that reminds me of sugar, sucrose binding proteins, which are in terms of molecular weight much larger structures. And judging from the binding energy you have obtained so far, around 8 kcals, I agree that that's not sufficient to actually maintain a good thermodynamic and thermodynamically stable coupling to the target molecule, in this case sucrose. So I think the approach you're taking by adding, for example, hydrogen bonding and perhaps salt bridges, which is exactly the same way proteins bind through non-bonded, non-covalently bonded interactions to the ligands and the ligands to the proteins, is the safe approach. Now, the number of atoms you have available in sucrose to actually do hydrogen bonding is relatively limited. How much energy do you think you can gain by adding the possibility of both hydrogen bonds and salt bridges to improve the thermodynamic stability of your ligand? Which, again, I think it's a very good approach. You're combining both binding and geometry to make the selectivity, to improve selectivity. But my main concern is really how much thermodynamic stability can you achieve at this point under physiological conditions to make sure we hold on to the sucrose.
Okay, thank you, teacher, for the question. And as one number that I have in my mind about the addition of one hydrogen bond, for example, I think that we could increase the binding energy by 1 kilocalorie mole. Yes, then okay, that's one possibility. In this case, any way that we can use to increase the binding energy is anyway is welcome. Yes, but I think that with the addition of electron drawing groups in the phenylboronic groups, then will improve even more the binding energies than the addition of one or two additional hydrogen bonds with the free hydroxyls. Then I don't know, probably if we got a binding energy about 12 kilocalories mole or 15 kilocalories mole, we will got enough binding energy to differentiate or get a high selectivity and stability for the detection.
Okay, yeah, I agree. I mean, 12 kcals would be a reasonably stable binding energy. Do you think you're going to have any issues in terms of sterics with your ligands, in terms of how flexible the molecule can be in the presence of other larger molecular weight structures?
Yes, and yes, but basically the flexibility of the ligand will depends on the molecular structure of the bridged atoms of the backbone that bridge both phenylboronic acids. That's one of the parameters that I should optimize. But with these initial structures proposed with the library of this overlay program, I found some rigid structures that are composed mainly by aromatic rings. Yes, and something more rigid than aromatic rings with carbon atoms. I think could be difficult to get, and I think this should be taken into account with the posteriori improves of these structures.
Great. So one low-hanging fruit that you might want to try. So definitely you need to look at the dynamics of these ligands to the sucrose in the solution, and basically do some molecular dynamics and see how stable they are. And the second one might be to add a fluorophore or a chromophore to your ligand as a first level approach to do some sort of detection of sucrose in solution. So instead of binding covalently the ligand to a surface like an electrode or some other device for which we would use, for example, field effect transport conditions, we could use fluorescence. And that might be interesting. Now, I'm going to couple that with a question we have from Nicolas. And Nicolas is asking how expensive would it be for actually developing these ligands through organic chemistry? And that's a, you know, it's a sensible question, because it's always a challenge to produce the ligands. So
yes, I that's the, I mean, this is one step after another, and my main task in this moment is try to get some molecular complete molecular structures. Then the next step, when I have some structures that means that for example, with binding energies about 15, 12 kilocalories mole, I will work in the retrosynthetic way to produce these structures. A retrosynthetic way means that we have I should break this molecule, this big building tail ligands with, okay, I have now the principal part, that is the phenylboronic acid. Phenylboronic acid, I don't have in this moment the prices of how many, which is the cost of some grams of phenylboronic acid, but it can be ordered on the internet from Sigma Aldrich. Then the principal component, it's easy. But something that I didn't mention in my presentation is that this overlay program also generates the library, the principal molecules used for the formation of the bridge, of the backbone bridge. And this information is useful. Just for lack of time on the presentation, I didn't show that, but we have that information, but I need to go deeper in this information in the next months.
Great. Again, thank you, Gustavo, for a good talk, and for the questions from the audience. We're going to have to move on now because we're slightly behind schedule now. So congratulations, virtual applause, Gustavo, and let's move on. We're going to have our final talk before our poster session. And this talk is going to be by Isabela Lopez. So Isabela is an electronic engineering student, last year student. She's working on her thesis developing a sensor for polysaccharides in solution. And again, this is important for measuring starch implants, and her approach is through field effect devices. So Isabela, we're all yours. Thank you.
Hola, soy Isabela López, estudiante de Ingeniería Electrónica de la Javeriana Cali, y les voy a presentar los avances de mi proyecto de grado bajo la dirección del profesor Andrés Jaramillo. Este trata sobre el diseño in silico de un sensor basado en un transistor FET de grafeno para la detección de almidón. Les voy a exponer entonces la motivación del proyecto, una breve explicación de los transistores de efecto de campo y el grafeno como material usado para esto. Luego les expondré el diseño, todavía en proceso, el nanosensor, la funcionalización del canal, la geometría y los resultados hasta ahora del transporte electrónico. Y para terminar, las conclusiones. Así como la medición de biomarcadores es de gran importancia en aplicaciones para la salud, como hacer el seguimiento de un paciente o detectar la de manera temprana una enfermedad o para detectar de manera temprana una enfermedad. De la misma forma, se usa para la detección de metabolitos en plantas. Uno de estos es el almidón, el cual juega un rol muy importante ya que permite hacer un seguimiento de crecimiento, almacenamiento de energía o indicador de cómo la planta se adapta a un estrés exterior. Por lo tanto, pues el monitoreo continuo de síntesis intracelular es fundamental para comprender las vías de energía y regulación metabólica en las plantas. Los métodos convencionales de medición son muy invasivos con la planta y pueden requerir de equipos o procedimientos externos para el análisis de la muestra. Sin embargo, pues para una medición en tiempo real de estos metabolitos es necesario un sensado in vivo y que sea capaz de detectar concentraciones muy bajas y capaz de indicar el estado de estas variables sin comprometer la salud del organismo vivo. Esto es lo que se propone con este proyecto. Se propone entonces un sensor basado en transistores de efecto de campo y pues los transistores de efecto de campo, el funcionamiento básico consiste en que se controla el flujo de los portadores, de la mayoría de los portadores de carga, sean huecos o electrones, desde la fuente hasta el drain, afectando las dimensiones y la forma del canal conductor en medio de ellos por medio de un campo eléctrico. Este campo eléctrico pues se genera a través de la aplicación de un voltaje de gate. Por otro lado, el grafeno pues es un material de 2D, de 2 dimensiones, que consiste en que átomos de carbono pues están organizados en forma de honeycomb. Esta hoja de grafeno, presenta propiedades electrónicas únicas, como lo es pues una alta movilidad, una baja dispersión de cargas, una alta conductividad, cero band gap, o sea, un comportamiento metálico. Sin embargo, pues si no quiere un comportamiento semiconductor en ese tipo de materiales, lo que se necesita es reducir sus dimensiones. Y el comportamiento electrónico queda descrito en función de los bordes, el tipo de borde de la cinta de grafeno. Entonces hay 2 tipos de borde, el armchair y el zigzag. Estas pueden ser metálicas o pueden ser semiconductoras dependiendo de el ancho de la cinta. En la gráfica derecha se presentan las diferentes brechas de bandas de cintas armchair según su ancho en número de átomos. Teniendo, por ejemplo, que la cinta que se muestra armchair en el medio tiene un comportamiento casi metálico con una brecha muy pequeña. Y por el contrario, si tuviéramos una cinta armchair de un átomo menos de ancho, pues tendría un comportamiento semiconductor. Bueno, ahora los transistores de efecto, el punto del proyecto es utilizar transistores de efecto de campo basados en grafeno. Eso es decir, que tienen el canal, el canal es una cinta de grafeno. Es importante eso porque pues el grafeno va a permitir que cualquier molécula externa se pueda posicionar en la superficie del canal y esto pues va a afectar las características electrónicas del transistor. Lo que utilizaríamos para medir la presencia o no del analito. Entonces, como mencioné antes, pues el interés de ese proyecto es la detección de almidón. En este pues este está conformado por dos tipos de polímeros de glucosa, la amilosa y la amilopectina. La amilosa es un polisacárido cuya cadena pues es de una forma lineal, y mientras que la amilopectina pues es altamente ramificada. Por esta forma que tiene la amilosa, pues se escoge como molécula objetivo pues del nanosensor, y por medio de ella pues se detecta la presencia de almidón. Listo. Se necesita entonces funcionalizar el canal para la detección específica de amilosa, y en la literatura pues se encuentra que se utilizan moléculas del tipo pirenos, se llaman, para funcionalizar los nanotubos de carbono o las hojas de grafeno, ya que no es una funcionalización no covalente, sino más como que el pireno se físico absorbe en el canal. Pues, por ejemplo, en específico, pues estos dos, pues se utiliza ahí el de la mitad, utiliza un pireno, pireno con ácido borónico en el extremo que reacciona con la molécula objetivo de forma covalente, y se utiliza este tipo de pireno para la detección de glucosa. Dicho esto, pues nosotros se utiliza un pireno también, que es pireno butamino, butamino. Dicho esto, nosotros utilizamos un pireno butanamido fenil con ácido borónico, porque pues se ha dicho en la literatura que ya se ha utilizado para la detección de azúcares simples, y pues la amilosa está basada, es una cadena de glucosa. Para corroborar y que la amilosa y el pireno, si que tienen la reacción, si se tiene entre ellos la reacción covalente y, y se mantiene estable, pues esa reacción, pues hacemos cálculos de energía de binding por medio de métodos ab initio, y pues nos da resultado de que nos da una energía de binding negativa, lo cual quiere decir que el amilosa sí prefiere estar ahí enlazada con el pireno. Bueno, aprovechamos entonces para la geometría del transistor, aprovechamos las características de las cintas tipo Armchair, que pueden presentar comportamientos casi metálicos y semiconductores, para hacer una única cinta de grafeno, y que ese sea prácticamente el transistor. Cambiando solamente los anchos de la cinta, podemos poner los dos electrodos a los extremos, y pues en el medio el canal semiconductor. Se evaluaron distintas geometrías del transistor, variando anchos de la cinta de grafeno y variando el largo del canal, y pero pues al final se llegó a esta configuración y no se hizo el canal muy corto, pues porque el transporte el canal es muy corto, domina el tumelamiento y ese no es el objetivo. Entonces aquí vemos el diseño del transistor completo y pues se hace el cálculo ya de corriente voltaje para el transistor completamente limpio, sin pirenos, sin amilosas, y se compara en esta gráfica de abajo con ya con el transistor, la característica de corriente voltaje con el transistor, con los pirenos, en el grafeno, sobre el grafeno. Se puede observar entonces que pues, los pirenos no alteran las características electrónicas del transistor, y esto es algo vital porque se garantiza que el cambio en estas características para detectar la amilosa va a ser independiente del ligando, que la única que, y es lo que nos interesa, que la única que cambie las características del transistor sea la amilosa y que se pueda medir ese cambio. Entonces, hacemos, ya una vez tenemos la amilosa B, ya tenemos los pirenos, procedemos a hacer los cálculos con una y dos amilosas. En esta y la anterior, y estos anteriores cálculos, como el de aquí de la derecha arriba, fueron hechos son cálculos utilizando métodos semiempíricos, los cuales son computacionalmente menos costosos, pero pues no son tan acertados. Hacen aproximaciones. Y pues se puede ver que no hay diferencia en transistor limpio, transistor con los pirenos y transistor con las amilosas. Y pues aquí es en donde nos encontramos actualmente en el proyecto, evaluando el por qué, cómo hacer de que se vea la diferencia en el cálculo de corriente voltaje para el pireno con las amilosas. Y pues, una de nuestras primeras decisiones es como ya no utilizamos los métodos semiempíricos, sino que pues comenzamos a hacer cálculos con ab initio. Pues por ahora, como se ve en el de abajo, no se ve mucha la diferencia con una amilosa, pero pues vamos a ver cómo de pronto no detecta una amilosa, sino que detecta desde dos amilosas. Y pues este cambio en el método pues se dio porque nos dimos cuenta que hay funcionales que describen muchísimo mejor el comportamiento de grupos químicos que, por ejemplo, que tengan el boro. Bueno, las conclusiones son que, bueno, usando una sola cinta de grafeno Armchair para los electrodos y para el canal, nos permite obtener un diseño del transistor simétrico, compacto y práctico. El grafeno, pues como un sensor, pues, o como material para hacer sensores, es adecuado para aplicaciones, muy adecuado para aplicaciones como esta, pues porque es un material altamente biocompatible, porque está totalmente pues, basado en carbono, y que el canal de este tipo con el grafeno pues nos proporciona un gran área de superficie que está la cual está directamente expuesta al ambiente de sensado, lo cual hace que todo el canal sea susceptible, pues con todo el canal sea posible sensar la amilosa, el analito que estamos interesados. Listo. Muchas gracias por su atención.
Okay, Isabella, thank you for your talk. Some of the graphs were not easy to follow because of the video quality, but hopefully the audience understood. So before we get into questions from the wider audience, I would ask you the following. So amylose is a polymer, right? It's basically a polysaccharide, and you're binding to a ligand that is non-covalently bonded to the graphene surface. So it's a small ligand that is bound through the aromatic rings of the pyrene moiety to the rings of the graphene. So the binding energy is probably somewhere around less than 10 kcals per mole, okay? Now then you bind to the other end of your ligand, to the open end, you're binding a polymer that it can have different degrees of polymerization. It can be a long chain, it can be a small chain. How do you think that's gonna, behave when you have a long amylose chain? That is, the molecular weight of that long chain is going to, do you think it's going to drive the pyrene out of the graphene pi-pi stacking, or do you think it's going to be stable?
So that I'm not sure if I understood, but you're asking me if when the more large chain bonds with the pyrene, then maybe the pyrene gets like out of the surface of graphene?
That's my question. Yes.
Okay.
So it would
well,
be.
I during the for example the thermodynamic simulations, so
Okay.
the molecular dynamic simulations, we did simulate how the pyrene with the amylose bonded interacted and like they behave when we put on some temperature to the system. And I remember that the amylose not like took off of the surface the pyrene, but the amylose tried to go around the graphene.
Okay.
So I don't know if the graphene, if the amylose could do that.
How big was your chain in that case? How many monomer units?
Not so big. Three glucose.
Okay.
Ring.
Okay. So a trimer. Okay. So I think it's important to do that sort of simulation just to make sure it's thermodynamically stable. And I think the approach you're using is good, meaning you have to do thermodynamic molecular dynamics calculations to prove that under physiological conditions and in solution, indeed your ligands are stable when they bind to the amylose. So I think that's a good sanity check. Okay. I don't know if we have any other questions for Isabella. If not, I'd like to thank all the speakers, including Isabella from this morning. A virtual applause to them. And we are now moving into the poster sessions. And let me just briefly share my screen with all of you so that we can explain the procedure here. So hopefully everybody can see. Now you will have to go to this URL to navigate through the posters. We're going to have a 30-minute session of posters between 10 and 10:30 before we resume our regular talks. So if you go to this URL, then you will land into this page. And then if you click start, then you should be able to navigate. Unfortunately, I'm not doing it on my computer, but on my iPad, and it might be a bit of a challenge. But anyway, you can now navigate through the setting of the university campus and look at the individual posters that you see in the session by clicking on the poster. Once you click on the poster, you should see the particular presentation and the live streaming from the authors of that poster. So again, you have 30 minutes to explore the different contributions, and I hope you enjoy the navigation. Thank you to Luisa Storquiza for his work on this virtual navigation system for the posters in the symposium. Anyway, so I'll see you guys in about 33 minutes back again into our regular talk sessions. And for now, enjoy the poster session. Thank you.
Bueno, muy bien. Continuamos. Okay, everybody. So we're going to continue on our regular schedule of talks after a brief attempt to navigate the virtual poster session. I was able to interact with two posters. I think for the next session or prior to the next session, we'll give a brief tutorial on how to navigate the system. Hopefully that will enable everybody to actually get a hold of the speakers and interact with them through the posters. Now, our next session or our next speaker is Harold Murcia. He's an electronics engineer, and he's got a master's in industrial control, and he's going to be speaking about the development of methods to understand environmental information through remote monitoring and remote sampling through robotic systems, UAV systems. So Harold, please take it over. Thank you.
Hello, my name is Harold Murcia, and I will be presenting our work developing a simulation tool for 3D plant modeling based on 2D LiDAR sensor. This work was funded by Omics P4 project and was done in collaboration with David Sanabria, Dairo Mendes, and our advisor Manuel Guillermo Forero at University of Ibagué. Plant phenotyping is a science that links genomics with the ecophysiology and agronomy of plants. The functional body of the plant, which we call phenotype, is formed during the growth and development of the plant from the dynamic interaction between the genetic background, which we call genotype, and the environment. Machine vision systems have been part of the digital analysis of plant phenotyping under different measurement principles: regular cameras, multispectral cameras, hyperspectral cameras, and thermal cameras, among others. However, although great advances have been made, these technologies have a two-dimensional representation of a three-dimensional reality. Under this principle, different works have emerged with the intention of conducting a three-dimensional analysis of plants. In this field, different authors have presented efforts in the phenotyping of three-dimensional plants, both in field and laboratory experiments and in simulation, given the different conditions that must be established to obtain satisfactory results. This work focuses on the 360 degrees modeling of plants by using a 2D laser sensor, a rotational disc, and a software model that coordinates the obtained signals from the perception subsystems with the movements of the action subsystems to transform the data into a single reference framework and to generate a three-dimensional model of a plant. In this way, we propose an open source development under a widely used software platform such as Robotic Operating System, ROS, and Gazebo for the simulation of 3D robotics platforms. Our system is composed of a 2D laser device, a step-to-step motor, and a rotation platform that supports the object to be scanned. Parameters such as rotation resolution and measurement resolution can be configured so that, firstly, the reconstruction algorithms can be validated before implementation, and secondly, artificial point clouds can be generated from the 3D plant models that can be obtained from different databases on the internet. But what is LiDAR technology and how does it work? LiDAR, or light detection and ranging, follows a simple principle. A laser beam is from the device to illuminate a target, and in response, a reflection is generated which is perceived by the same device, so that it's possible to measure the time elapsed from the shoot to the reading, and then determining the distance between device and target with a simple equation. A two-dimensional laser adds a motor with a rotating mirror which allows to illuminate different areas of a scene. So you get different measurements depending on the angle of the motor. Our design started with the mechanical configuration of the system in SolidWorks, which was migrated to Gazebo Simulator. Then a generic kinematics model was developed and programmed in Python language under the ROS system to generate a raw file with the data of all the measurements according to the configuration parameters of the simulation. To motivate our work, I want to show a short video of our process and results. Finally, a second computer model transforms the raw data into a XYZ model and saves it in TXT format, which can be easily transformed into other formats such as LAS, PCD, or others. Thank you for your attention.
Okay, Harold, thank you for your talk. I understand there was a misunderstanding on the length of these talks, so some of them are shorter than expected. So Harold is going to add some slides and discussion beyond the video. Go ahead, Harold.
Thank you, Professor Andres. Can you hear me?
I can hear you, yes, go ahead.
Thank you. And do you see the presentation?
Yes.
Thank you very much. So just for complementing the previous presentation, I would like to show you some details with respect to the project. So the idea, as I showed you, was to generate a three-dimensional model or representation of a plant previous to the real implementation, I mean with simulation. So basically, from the engineering problem, we have 2 points with a LiDAR sensor, which is centered in P1 with its own reference frame. So the idea is to transform all the collected data from in the LiDAR to centered and unique feature frame, which is centered in P2. So we have to collect all the information and transform them to accumulate and generate a three-dimensional model. So for this problem, we found a solution based in a transformation with 2 moments. The first one is a translation. And in translation, we have 3 parameters which represent the distance from P1 to P2 in each axis. I mean X, Y, Z. And the second moment is focused in the rotation of the platform. So basically what we need is a measurement of the rotational disk or estimation, depends on the system, to have the angle of the rotational disk and then have a rotational matrix to then have 2 moments of transformation and generate a final equation, which is the equation 3, which transform information from P1, accumulate and generate the three-dimensional model the full way, I mean the 360 degrees. So the plant in this case is on the rotational disk P2, and then we perform an experiment to have different measurements and then generate the three-dimensional model. So to validate results and simulation, we choose 2 plants, 2 models, a wheat plant and a corn maize plant. And then we define 3 parameters for each plant, the height, individual stem diameter, or stem distance, with the idea to generate an index error based on this simple equation. When we have the measurement obtained from the simulation and the reference value, which is a reference value taken from the ground truth from the real 3-dimensional models. So we performed the experiments, two experiments for each plant in simulation, and we made experiments at 2 resolution. I mean, two different steep degree steps. So the first one was 1 degree step, and the second one was carried out at 0.5 degree step. So then we have a reconstruction, a 3-dimensional reconstruction, a 3-dimensional model for each plant, and have the measurements to compare with the reference value. So in the first case, the general or the average error is around 20% at a 1 degree step resolution. For the second case, we have around 5%, which is a big change from 20% to 5% error. Just we change the resolution step of the platform, I mean the step of the rotation. For the other experiment with the second plant, which was corn, we have the same experiments at 1 degree step and 0.5-degree steps. And we have also a big change, improving from around 7% to 6%, more or less. Was not a big change like the previous case, because the first plan had more detail, had more high frequency information, so higher resolution is needed. It's not the same case for the plant, for the current plant, but however, there was an improvement on quality of generated three-dimensional model of the plant. So once we have these results, I would like to share with you some conclusions. This presentation describes a work in progress. At this moment, we are working to migrate from simulation to real system, the development of a software tool to simulate the 3D plant modeling process, which can be easily migrated to real application under ROS frameworks. So Altitude is a simulation. What we present here is also the possibility to migrate this software to real implementations in different possibilities. Our source code implementation is freely available online and can be obtained from this link, which is also on the paper. And well, we invite you if you want to install on your computers or your systems. Computer simulation environment brings an approximation to real scenarios in three-dimensional reconstruction systems, allowing to validate reconstruction algorithms and to generate artificial point clouds of plants for its study. Some parameters such as step resolution, like LiDAR resolution, which is the LiDAR distance or device resolution, have a very big impact on the quality of the generated models. Also in the size of the point cloud, the number of points, the computational cost, and of course the simulation time. More resolution needs more simulation time. In our future directions, will involve to build plugins for simulate other parameters in addition to the range or the distance, because the laser sensors have in the response more information like intensity or like pulse width, which is also very important for these kinds of applications. Other future direction is integrate automatic estimators for an automatic tuning in function of the parameters of the models, and then migrate to the real application, as well as migrate the simulation to different real scanning platforms. Well, thank you to Omics, which is the work was funded by the project Omics and Optimization Multiscale in Silico de Cultivos Agrícolas Sostenibles. And well, thank you for your attention and for your time.
Thanks, Harold, for supplementing the video. Very interesting. So I have a few questions. Let me see if we have any other questions on the board. We don't. So let me go ahead and ask them. So it seems to me that you basically did a code, or you wrote a code to model the scanning, the three-dimensional scanning of a plant, a fixed on a rotational base. And for that, you did spatial transformations, translation, rotation. I'm not sure if you included perspective. It seems that you did not consider perspective in the transformations as part of the transformations. But it's easily incorporated. You could express the spatial transformation with a simplified notation or a simplified transformation set, such as the ones from the typical robotics field, in which you use a Denavit-Hartenberg type approach to have a single matrix operator that does rotations, translations, and scaling, and perspective. So with a single 4x4 matrix, you can express all 4 transformations. So that's you know, an initial suggestion. I mean, there's no expense computationally speaking in your case because the number of degrees of freedom is really not high. Now, if you're going to develop a model that is supposed to describe a physical system, such as a plant, right? And you built this apparatus that you could take with you to the field to actually do a 3-dimensional scan with a LiDAR system. Are you planning on incorporating things like the dynamics of the plant? I mean, there's actual motion of the plant that will give you errors in the scanning that you need to compensate. So the leaves are moving, you know, it's hard to keep a plant completely still. So what is the expectation when you connect this simulation tool to a real system? And again, within Omicas, if you talk to Professor Colorado in Bogota, probably your professors at Ibagué, there are options to actually do the physical LiDAR experiments. So what's your plan right now?
Well, thank you for your question, Professor. Well, the first one, with respect to the scanning and perspective, we presented as just a simple solution. I mean, simple as possible, which is translation and rotation. But of course, by including scaling or perspective, it's possible to improve the quality and the error obtained with these possibilities. That's the idea of simulation, because we can identify problems previous to the real implementation. So yes, in our next future direction, we have also the intention of include perspective and scaling, and also to include automatic tuning algorithms to define optimally the parameters of the model, which have a very big responsibility of the total error. With respect to the dynamics, that problem can be solved by improving the frequency of the sensors. I mean, if you have a low sample frequency, you have more problems with the dynamics, with the movements, with the wind or light conditions. But if you have a faster system, dynamics problem is not so important. So it depends also of the sample frequency, and also by including dynamics models like pose estimation or for instance go on a step. It's a technique, so you move and you wait a moment for a steady stable, and then you have a measurement. There are possibilities, but we have to test in real conditions. In our future direction, we have in mind to migrate our simulation to a real platform and then integrate pose estimation models and dynamics consideration, as you mentioned.
Okay, yeah, I agree. I mean, the dynamics of a plant are really not very high frequency. So if you actually calculate the highest frequency of motion in your plant and you apply the Nyquist criterion, then you would know what the sampling frequency should be, which is double the maximum frequency of your vibrations in the plant. So I don't think that's a difficult problem, considering it's a very slow motion system. So in terms of going to actual experiments with a LiDAR, how far are you guys along the way? Because I think 3-dimensional characterization of plant architecture is important when doing phenotypic analysis and when you are interested in relating the genotype with the phenotype. And you know, I just want to know how far along are you guys in that process?
Well, at the moment, we have some results with our new systems. We have 2 real systems. The first one was based with a very simple laser, which is a low-cost system. Results are interesting, but are not so good. We have tested also with Kinet, which is a popular solution in this case is a device from video games from Microsoft corporations, but it's very used in different applications in robotics or in this context of remote sensing and in laboratory conditions. We have very good results in comparisons, and recently we achieved better sensors. I mean, with high resolution, both sample frequency and angular step. So a better sensor from SICK company, which is a very, very popular LiDAR manufacturer in this context. So at the moment, we are in implementation of the total system with the last sensor. And I think we are close to obtain models in real-time conditions. And also our idea is include cameras to include color, like red, green, and blue cameras, and include color to these point clouds. So at this moment, we have only X, Y, Z information, just to calculate morphological parameters. But once we have color information, it's possible to have more possibilities in the point cloud and its applications.
Okay, great. So we have a question from Professor Teran. The question is, many physiological aspects of plant growth architecture and fitness depend on what's happening in root systems. How do you think you can integrate these important aspects into your model, perhaps in the future? Is that possible at all?
Well, roots are a big challenge. I mean, from the point of view of methods, it's basically the same, but you need more resolution. Depends of the size of the roots. So it's a big challenge from the point of view of instrumentation. I mean, perception or the resolution of the sensors. So it's possible. Yes, we have two problems. I mean, the first one is, as I told you, the resolution of the sensors because of the little size of the roots. But if we have a very, very good sensor, very, very high resolution, it's possible to obtain information or a model representation of roots. The second one is the occlusion. Occlusion is because we have shadows. In this case, we have a root behind other roots. So it's difficult to see how is the morphology of that element behind other. So the problem is occlusion because of the shadows. So in that case, there are some studies by adding wind elements like motors or fans to try to discover what is behind of the roots. So I think it's possible in the future, yes, but then we need to work in two ways. The first one is to improving the resolution of the sensor. And the second one is to define methods or model to solve the problem of occlusion.
Okay, Harold, thank you very much. Very, very complete response. And thank you for the audience for for the questions, in particular, Professor Teran. So we're going to move on. Virtual applause for Harold, and we're going to move on into a different topic, nonetheless important in plant science. And this talk is from Manuel Valencia. He's going to be the speaker. Manuel is working on the use of NDVI, that's normalized differential vegetation index, to predict the content or the emission of nitrous oxide from sugarcane crops. Manuel, please take over.
Buen día. Mi nombre es Manuel Camilo Valencia Molina. El día de hoy vengo a presentarles unos avances acerca de una investigación relacionada al uso o el potencial uso del índice de vegetación normalizado como una herramienta para estimar las emisiones de óxido nitroso en cultivos de caña. El equipo de investigación, el equipo de trabajo en el desarrollo de esta propuesta, ha participado Catalina Trujillo, Sandra Loaiza y Brian Múnera. Ellos hacen parte de las instituciones que cooperan o instituciones aliadas para el desarrollo del programa Omics. La presentación la hemos estructurado en 3 componentes. El primero es una introducción, el segundo una descripción de algunos materiales y métodos y el tercero una presentación de algunos avances de resultados. En esa introducción nosotros hemos empezado por responder ciertas preguntas. La primera de ellas es ¿por qué el índice de vegetación normalizado? Y esto básicamente es porque es un modelo matemático que nos permite determinar la cantidad, la calidad y el desarrollo de la vegetación a partir de la relación entre la absorbancia y la refractancia de bandas del espectro electromagnético de una superficie. Entonces, ¿qué quiere decir esto? Nosotros hemos buscado fuentes de información o bases de datos en Internet, fuentes libres de libre acceso, en las cuales hemos podido obtener imágenes con una muy buena resolución espacial que son capturadas generalmente por sensores de satélites, están adaptados a satélites. En ese sentido, el índice de vegetación normalizado es uno de los índices más utilizados en el análisis de coberturas y a su vez también en la aplicación en agronomía. Tiene múltiples aplicaciones en agronomía y hay algunas modificaciones, sin embargo es un índice muy popular. ¿Qué sucede? Que resulta que dentro de uno de los objetivos es que dentro del análisis de los procesos de emisión o de los flujos de emisión de óxido nitroso en sistemas de producción de caña, por la escala de la producción, por ejemplo en el Valle del Cauca, por la escala de la producción, nosotros lo que buscamos es poder hacer un análisis regional que nos permita tener un poco más de claridad o ser un poco más concreto en las estrategias o en la identificación de los principales factores que afectan estos procesos, debido a su importancia o su relevancia desde el punto de vista ambiental y en aras de la sostenibilidad productiva. Bueno, como les decía, entonces el índice está en función de algunas bandas del espectro electromagnético, principalmente el infrarrojo cercano y el rojo. ¿Okay? El modelo matemático que lo describe es el que ustedes pueden ver aquí en pantalla, donde es infrarrojo cercano menos el rojo sobre infrarrojo cercano más el rojo. En ese sentido, yo les mencionaba, utilizamos una fuente de información en línea, que fueron las imágenes del satélite Sentinel-2. Más adelante vamos a describir un poco más qué es este satélite, y en el que él se caracteriza porque tiene 12 bandas, tiene sensores para 12 bandas espectrales, entre ellas la del espectro visible, las que son percibidas por el ojo humano. Entonces, yo les decía, se obtiene o se trabaja a partir del rojo, de la banda del espectro del rojo y del infrarrojo cercano. Y se logra hacer se desarrolla el índice y los productos generalmente se presentan de esta manera, en el cual se trabaja en una gama de rojos a verdes, donde en la mayoría de los casos, entre más verde o mayor sea la intensidad en el verde, quiere decir un mayor desarrollo en términos de vegetación, y entre más esté aproximado a ese rojo, quiere decir que está relacionado con suelos con baja cobertura o incluso suelos descubiertos. Bueno, entonces ahora, ¿por qué? La segunda pregunta que nos hacíamos era ¿por qué óxido nitroso? ¿Sí? Entonces, el óxido nitroso hace parte de los 3 principales gases de efecto invernadero que provienen de los sistemas de producción agropecuarios. Tiene 3 características fundamentales. Una, que tiene la capacidad de absorber 310 veces mayor radiancia infrarroja en relación a la molécula de referencia, que en este caso es el dióxido de carbono. ¿Sí? La siguiente es que es una molécula que puede o tiene la capacidad de degradar el ozono estratosférico, ya que puede reaccionar con el oxígeno monoatómico que se genera durante el proceso de fotólisis del ozono. El tercer aspecto es que esta molécula puede estar estable en la atmósfera hasta por 114 años. Es decir, esto de cierta manera es un periodo, una ventana de tiempo muy grande, que lo que le permite es tener un amplio número de posibilidades para reaccionar con ese oxígeno monoatómico que les mencionaba hace un momento. Esta hace parte de una representación, es una molécula que no es simétrica, es decir, porque es un enlace, son dos átomos de nitrógeno unidos a un oxígeno. A diferencia, por ejemplo, del dióxido de carbono, que el carbono está en el centro de la molécula, de tal manera que queda un oxígeno a la derecha y un oxígeno a la izquierda. En este caso no hay simetría. Bueno, entonces, otro aspecto que nosotros le damos a la relevancia del óxido nitroso es que cerca del 27% de las emisiones globales provienen de origen antropogénico, es decir, de origen de actividad humana. Y de esa emisión antropogénica, es decir, ese 27% de las emisiones globales, el 80% está relacionado o es debido al uso ineficiente de fertilizantes nitrogenados. En este sentido es donde nosotros vemos por qué la relevancia del óxido nitroso y por qué su impacto o por qué su análisis en función de la producción agropecuaria de un cultivo como la caña de azúcar. La siguiente pregunta que nosotros hacíamos, en efecto, era, bueno, ¿por qué caña de azúcar? ¿Sí? Y es que a nivel mundial la caña de azúcar es un cultivo ampliamente, que tiene amplia expansión en la zona intertropical principalmente. Hay reportes también en la zona subtropical, pero casi toda la producción está concentrada en la zona intertropical. Y en 2018 se reportaron aproximadamente 27.7 millones de hectáreas sembradas en todo el mundo. ¿Sí? De esas, en 2019 se pudo reportar que en Colombia se sembraban aproximadamente 500.100 hectáreas, y de esas 500.100 hectáreas, 250.000 están registradas o se reportan en la región del Valle del Río Cauca, que tiene otra particularidad, y es que en esta región el modelo de producción de caña se hace bajo un concepto de alto nivel de tecnificación y bajo un modelo muy intensivo, en el cual se recomienda la aplicación generalmente de más de 100 kilogramos de nitrógeno por hectárea cultivada. Sin embargo, muchas veces estas recomendaciones o estas aplicaciones se hacen sin tener en cuenta ciertos aspectos técnicos, ciertos criterios técnicos que nos permitan hacer uso eficiente del fertilizante. Y culturalmente, lo que vemos es que muchos productores optan por hacer aplicaciones, aplicar más de lo requerido o de lo recomendado, bajo la premisa de que entre más nitrógeno se aplique, la planta va a responder mejor. Es decir, a partir del hecho o del tema de que es mejor que sobre y no que nos falte fertilizante. Bueno. Ya entrando en el desarrollo de las condiciones experimentales, nosotros trabajamos en la región, como yo les mencionaba, en la región del Valle del Río Cauca, en área rural del municipio de Cerrito, está en la zona centro del Valle del Río Cauca. Y nosotros utilizamos, o el periodo de muestreo de análisis que tuvimos fue del 20 de agosto de 2019 al 30 de marzo del 2020. Teniendo en cuenta que tuvimos muchas limitantes debido a todo esto relacionado a esta crisis relacionada a la pandemia, tuvimos unas ventanas en las cuales no pudimos hacer muestreos de manera tan intensiva. Y la información, nosotros después de que la obtuvimos o que la gestionamos en campo y que la gestionamos en la red, nosotros empezamos a hacer los procesos a través del software QuantumGIS. Yo entiendo después pudimos determinar que existen herramientas mucho más eficientes, pero en el momento fue con lo que iniciamos el análisis. A futuro, dentro de las recomendaciones está empezar a explorar el uso de aplicativos web que tienen mayor eficiencia y mayor capacidad de respuesta para el análisis a escala regional. Bueno, el tema de la información relacionada con la emisión acumulada de óxido nitroso, entonces nosotros lo que hicimos fue unas mediciones directas en campo, utilizando la técnica o utilizando los equipos, el equipo de una cámara estática manual, que se caracteriza en este lado. Pueden ver la cámara es un equipo bastante sencillo. Aquí en esta está desensamblado, es una representación de una cámara desensamblada donde tenemos una base, que la base básicamente es un cilindro, el cual nosotros vamos a enterrarlo hasta la mitad en el suelo. Es decir, nosotros vamos a insertar esta primera parte o esta primera mitad en el suelo y vamos a dejar la otra mitad de área libre, por así decirlo. Y a esa cámara lo que nosotros hacemos es que le acoplamos la tapa y con esta banda negra, esto es una banda de goma, nosotros lo que buscamos es de cierta manera fijar la tapa y contribuir a que se disminuyan las pérdidas de gases por la unión, en la unión, ¿no? Entonces nosotros ahí generamos una condición, un microclima diferente, una condición atmosférica ahí dentro de la cámara diferente que nos permite de cierta manera, o a partir de esta, vamos a poder inferir cuál es la tasa de respiración del suelo y cuáles son esos volúmenes, esa concentración tanto de óxido nitroso y otros gases que se liberan en la superficie del suelo dentro de la cámara, y esto poderlo extrapolar a condiciones o a unidades fuera de la cámara. Para posterior a los muestreos, nosotros obteníamos 4 muestras en cada muestreo por cámara, y esas muestras las analizábamos en laboratorio a través de técnicas de cromatografía de gases. Posterior al análisis, nosotros lo que hacíamos era hacer unos cálculos empleando el modelo de gases ideales, la ecuación de gases ideales, y lográbamos determinar cuál era a partir de la concentración del gas, cuál era el flujo o la cantidad de gas que se emitía en determinado periodo de tiempo por unidad de área. En cuanto a la información multiespectral o la base de datos que utilizamos, yo les mencionaba, nosotros utilizamos la base de datos de la Agencia Espacial Europea, haciendo uso de los insumos recopilados en la misión Copérnicus, que la misión Copérnicus es la que contempla algunos de estos satélites, en este caso el satélite Sentinel-2. Nosotros, ¿qué hemos visto? Que hay aplicaciones, por ejemplo, del satélite Sentinel-5 que permiten hacer análisis o que permiten cuantificar los flujos de emisión o la concentración de algunos óxidos de nitrógeno en la atmósfera. Sin embargo, estos compuestos no incluyen el óxido nitroso. Entonces nosotros lo que buscábamos también era tener la mejor resolución posible a partir de esta información libre, y esa no la ofrecía el sensor Sentinel-2. El sensor Sentinel-2, cabe resaltar que tienen las bandas con las que nosotros trabajamos tienen una resolución espacial de 10 metros por 10 metros cuadrados en cada píxel. Es decir, que para efectos de esta información, estos datos, era aceptable. Y la resolución temporal de la captura de las imágenes era de 5 días. Cabe resaltar que muchas veces por condiciones de objetos extraños en la imagen, es decir, nubes o sombras, no se podían utilizar. Sin embargo, esto es una herramienta que tiene una resolución temporal relativamente buena y nos permite tener mucha información disponible. Listo, ya pasando acá al tema de resultados, entonces nosotros partíamos del hecho de mirar cuál era la dispersión o cuál era el comportamiento de los datos de la emisión a través del tiempo. Nosotros lo que miramos era la emisión acumulada en un periodo de 210 días en 2 zonas. El área de estudio nosotros la dividimos en 2 zonas de tal manera que pudiéramos tener un escenario contrastante. El criterio para separar estas dos zonas fue la conductividad eléctrica aparente. Y porque la conductividad eléctrica aparente es una variable que nos permitía, o contábamos con los equipos para hacer una caracterización homogénea en todo el terreno relativamente rápida, y esta variable está, según la literatura, está relacionada, pues tiene su relación con el contenido de humedad en el suelo, las texturas, hay una relación directa con las texturas arcillosas, y hay una relación directa también con los contenidos de sales en el suelo. Entonces, nosotros de ahí podíamos inferir ciertas características del suelo. Entonces, lo siguiente era mirar el comportamiento del índice, o sea, calcular el índice y mirar su comportamiento en el mismo espacio de tiempo. La primera situación se nos presenta aquí porque las imágenes o la información espectral disponible en muchos casos o generalmente no coincidía temporalmente con los muestreos de gases. Es decir que el muestreo de gases se realizaba un día y la captura de información espectral más cercana o el paso del satélite por esa zona no concordaban en términos temporales. ¿Esto qué sucede? Que es que la emisión de este gas particularmente es un fenómeno altamente variable, y al no tener esa congruencia temporal, nosotros lo que teníamos era mucha fuente de error relacionada con el ejercicio. Además que no podíamos relacionarlo fácilmente, ¿no? Era un poco más complicado establecer modelos de regresión o comparaciones uno a uno con esta información. Entonces, ¿qué hicimos? Lo que hicimos fue buscar una estrategia para llenar estos espacios en blanco, estos espacios sin datos, y la mejor estrategia que hicimos fue los modelos de regresiones, y probamos o hicimos un análisis de modelos de modelo lineal y modelos no lineales, donde veíamos que el modelo con el mejor comportamiento en las dos zonas, el modelo con el mejor comportamiento era el cúbico, que alcanzaba a tener un R cuadrado por encima de 8, y con un valor, con un nivel de significancia muy bueno. ¿Qué sucede? Que es que debido a la tendencia, nosotros no alcanzamos, debido a las condiciones tanto climáticas como logísticas y demás, nosotros no alcanzamos a llegar hasta el final del ciclo prosayectivo con la captura de información. Y nosotros, la literatura nos indica que el NDVI, el comportamiento del NDVI a través del ciclo productivo se describe más fácilmente por un modelo cuadrático que por un modelo cúbico. Entonces nosotros optamos por utilizar el modelo cuadrático para hacer estos procesos de interpolación, teniendo claro que, o partiendo del hecho de que también habían unos márgenes de calidad en términos del modelo bastante aceptables. En ese sentido, entonces nosotros lo que hicimos fue desarrollar dos modelos, porque veíamos que, si bien nosotros en las gráficas lo que vemos es que el comportamiento tanto de la emisión como del índice de vegetación no tiene un comportamiento o unas tendencias relativamente diferentes en función de la zona. Entonces nosotros lo que hicimos fue desarrollar un modelo para la zona 1 y un modelo para la zona 2, un modelo de regresión del índice en función del tiempo. Y este es una representación de cómo queda el modelo, y lo que veíamos hace un momento tiene unos niveles de un R cuadrado relativamente bueno, ¿sí? Y con unos niveles de significancia muy buenos. A partir de este modelo lo que nosotros hicimos fue llenar esos espacios y poder completar esas observaciones en términos de emisión y índice espectral de vegetación normalizada. Otra cosa que nosotros veíamos era que, bueno, teniendo ya el índice y teniendo los datos de emisión, empezar a mirar si se podían correlacionar estos dos valores, ¿sí? Entonces, el comportamiento, nosotros hicimos pruebas con diferentes modelos o coeficientes de correlación, y al final el coeficiente de correlación de Spearman fue el que tuvo los mejores resultados, teniendo claro que esto, pues, de cierta manera no es tan aceptable, pero una correlación del 0.75 pues ya empieza a ser un poco más adecuada. En ese sentido, nosotros lo que hicimos fue correr unos modelos en el que nosotros hicimos primero, lo que tuvimos que hacer era que debido a la naturaleza de los datos, era un poco más fácil de analizarlos si los transformábamos. Y lo que buscamos fue hacer una transformación del logaritmo en base 10 para poder determinar o para poder calcular el logaritmo en base 10 de la emisión de óxido nitroso acumulado en función del logaritmo en base 10 de del NDVI en las dos zonas de estudio, ¿sí? Igualmente, aquí nosotros lo que veíamos era que los modelos, el modelo de mejor ajuste era un modelo cúbico y el modelo cuadrático pues ya era el segundo modelo con mejor ajuste. ¿Qué pasó? Que en este caso nosotros sí optamos por utilizar el modelo de mejor ajuste debido a que ya tenemos, pues estábamos buscando los niveles, los datos o los modelos con mayor grado de significancia y mayor correlación, ¿sí? Que describieran un poco mejor el fenómeno. Y debido a que no tenemos antecedentes hasta el momento del uso de índices de vegetación normalizado para relacionarlo con las emisiones de óxido nitroso, pues no sabíamos cuál era, no teníamos un referente de cuál podría ser la tendencia, por eso nosotros optamos por utilizar el modelo cúbico en las dos zonas. ¿Qué sucede? Entiendo que todavía en los modelos la información necesita ajustarse mucho mejor, necesitamos tener unos niveles de significancia un poco mayor, o no de significancia, sino una mayor relación en el desarrollo de estos modelos, un coeficiente de determinación mucho mayor, pero esto sí nos abre una puerta para explorar o para poder mejorar el desarrollo o las estrategias para el análisis a escala regional de las emisiones de óxido nitroso a partir de imágenes multiespectrales. Porque si bien nosotros, este es nuestro ejercicio preliminar, por así decirlo, ahora el siguiente paso es empezar a validar ya el ejercicio y tratar de desarrollar un índice propio de las emisiones de óxido nitroso, que podemos estar en función de la adaptación de índices o la relación con índices ya desarrollados, o podemos también empezar a explorar el uso de las bandas por separado, de las diferentes bandas, saber cuál es el comportamiento del modelo del fenómeno en función de estas bandas. Bueno, eso era básicamente lo que tenía para contarles. Entiendo que se generan muchas preguntas, muchas dudas y todavía estamos en un nivel de exploratorio, pero bueno, esa era la idea de nuestro ejercicio preliminar. Muchísimas gracias. Agradecerle también al programa Omics y a las instituciones aliadas que han contribuido al desarrollo de esto.
Okay, great talk, Manuel. Thank you very much. Really interesting. So, while we get questions, let me shoot one at you. And the question is mainly associated with the molecule itself, with N2O. So if you look at N2O, it is a symmetrical molecule, right? Linear. And it has three main vibrational modes. One at around 600 wave numbers, and one around 2200 wave numbers, and the other one I believe was somewhere around 13 or 1400 wave numbers. The highest intensity peak you'll get at around 500 wave numbers. Okay, and it's an interesting approach that you're using the NDVI metric, which is a near infrared index that consolidates pretty much all the absorption spectra of those three vibrational modes that you see in the molecule. How do you expect, or which one of those modes would you expect dominates in improving the correlation that you currently have from the NDVI to to the actual presence of N2O?
Buenos días.
Hola, Manuel.
Inicialmente, pues, trabajábamos el óxido nitroso, porque en algún momento nosotros veíamos que era un ejercicio bien, era como el índice más utilizado. Sin embargo, ahorita nosotros lo que necesitamos es explorar el uso de las demás bandas, ¿sí? Empezar a encontrar otras interrelaciones. No necesariamente tendrá que ser el NDVI, puede ser otro índice o otras bandas en las que trabajemos. Sin embargo, también nosotros estamos pensando en el uso de alternativas o modelos un poco más robustos en términos de análisis con relación a los modelos computacionales, posiblemente.
Okay, so I think it's a good, interesting approach. My question is, currently the NDVI usually can be captured through multispectral cameras, right? What is the range of frequencies that multispectral camera can capture in order for you to overlap with the vibrational modes that you're interested in N2O? Again, probably you won't be able to capture all of the three modes, the primary modes in the molecule. And you want to be able to capture at least the highest intensity modes to make it to a high correlation between an NDVI type index or another, basically some vibration, some deeper analysis of the vibrational modes. So that's my concern, that multispectral cameras might not reach the frequencies that you need to actually overlap entirely the vibrational modes of that molecule. But it's a very interesting exploration.
Bueno, para la segunda fase del ejercicio, lo que contemplamos ya es el uso integrar el uso de drones en los cuales podamos controlar esa frecuencia y podamos tener mayor disponibilidad de información, ¿sí? Con una ventana de muestreo un poco más corta. Adicional a eso, las unidades aéreas no tripuladas, los drones, permiten solucionar el tema, por ejemplo, de lo que yo les decía, los artefactos o las nubes o las sombras que se generan en estas imágenes capturadas por satélite. Es un factor que uno puede, como nosotros con un dron estamos volando en una altura muy por debajo de estas capas de nube, no generamos o tenemos mejor calidad en términos de imágenes, ¿sí?
Okay.
Let
me interrupt because we have 3 additional questions and very little time. So we have a question from Nicolas. He's asking, what is the reason for choosing the Spearman correlation coefficient? And in the same line of questioning, have you tried other ranked-based coefficients, for example, the mutual rank coefficient?
Okay. Bueno, la primera, yo les decía que el ejercicio para la primera pregunta que habla sobre el coeficiente de correlación de Spearman, nosotros lo elegimos porque fue de acuerdo al análisis preliminar de los datos. Nosotros veíamos que las tendencias en los resultados no se comportaban de una manera, con una estadística normal, por así decirlo. Entonces, habían modelos o correlaciones como Pearson, pero que son recomendadas más para ejercicios con datos normales. En este caso, una buena alternativa que nos ofrecía, o pues a partir de lo que revisamos en la literatura, era el modelo de Spearman, ¿sí? Y respecto a la... ¿Señor?
No, that's fine, go ahead.
Y respecto a la... que si hemos probado otros coeficientes, bueno, no. Básicamente, estamos en lo que yo les decía, estamos en un ejercicio exploratorio. Nosotros terminamos de hacer muestreos en campo, todo el ejercicio de muestreo lo terminamos hace 3 meses, y si bien hemos estado haciendo o hemos culminado con la fase de análisis en laboratorio, nosotros todavía tenemos los datos, todavía estamos haciendo el perfilamiento pues, de los datos para ver qué nos está sirviendo y qué no nos está sirviendo, ¿sí? Yo les mencionaba al inicio, la idea es como abrir la expectativa a una nueva posibilidad en términos de análisis, pero aún estamos necesitando mejorar las condiciones o el nivel de detalle, tanto en la recolección de información como en los procesos de análisis, poder afinar un poco mejor esto.
Okay, now there's another suggestion from Gustavo Lara. He's asking have you considered the use of real gas equations to improve your model predictions?
Bueno, no, en términos generales, nosotros hemos utilizado el uso de los modelos de gases ideales porque ya hay como una metodología establecida para el desarrollo o el análisis en términos del flujo de emisión, ¿sí? Nosotros con las cámaras lo que hacemos es determinar la concentración de esa atmósfera que se genera dentro de la cámara. Posterior a eso, nosotros lo que necesitamos determinar es ese flujo, pero en términos de miligramos. En ese sentido es que nosotros utilizamos el modelo de gases ideales, ¿sí?
Okay.
Ya para continuar con el análisis, bueno, ese modelo lo utilizamos porque es lo que la literatura nos ha mostrado, ¿sí? Que es la metodología por excelencia. Y ya para continuar con el análisis, si no hemos visto o todavía no tenemos claridad cómo podríamos integrar ese análisis al modelo. Nosotros ahorita estamos netamente en ejercicios de relacionarlo con el tema espectral, ¿sí? Hay unos ejercicios o hay unos modelos desarrollados, unos modelos computacionales que integran otras variables agroclimáticas y le permiten, de cierta manera, hacer operaciones similares que le permitan inferir cuál es esa emisión.
Okay, so Manuel, thank you. We have to move forward with the final talk of the morning. There's a quick practical question from an anonymous attendee. So basically it's associated with the estimation of the volume in the chamber of that, the camera that you place in the field. And if you actually rotate this camera periodically in order to have some statistical relevance in the volume of your chamber.
No escuché bien, qué pena.
So let me ask it in Spanish. So la persona pregunta que cómo se estimó el volumen de la cámara en campo y si tienen que cambiar la cámara o rotarla de alguna forma para garantizar medidas estadísticamente relevantes en tiempo o solo hacen un muestreo, por ejemplo.
No, nosotros hicimos el muestreo de acuerdo a las condiciones de manejo. Bueno, esto hace parte del tema que vamos a tratar o que voy a presentar el viernes, pero de acuerdo a las condiciones de manejo, nosotros conocemos las dimensiones o calculamos las dimensiones de la cámara porque están ya preestablecidas, es decir, a partir de radio.
Claro, pero la entierras en el suelo y no tienes la altura con respecto a la superficie del suelo, que es irregular también, ¿no?
Nosotros controlamos la profundidad de la enterrada, por así decirlo. La cámara, la base de la cámara tiene 20 centímetros de alto y nosotros enterramos 10 centímetros, ¿sí? A partir de eso le sumamos la altura
de
la...
en el suelo entonces para que quede bien homogéneo y una superficie fina, pues. Listo.
Exacto.
Muy bien. Bien. Manuel, tenemos que seguir. Muchas gracias de nuevo. Un aplauso virtual. Buen trabajo. Okay, so we're moving into our final stretch of the morning. Our last talk is going to be by Professor Jose Hernandez from the University of Ibagué. And his work is mostly associated with image processing, plant phenotyping, and precision agriculture. So please take it over, Jose. Thank you.
Hello, everyone. Thank you all for attending this presentation. in this virtual symposium in plant omics science, which is related with RGB imagery and also a deep resolution strategy for grain yield assessment in wheat. This experiment was carried out in collaboration with the Universidad de Ibagué, Itasil Institute, INIA Institute, University of Barcelona, and also supported by the Fellowship Formación de Talento Humano. In the groups, we have the DEMASTEC group in the Universidad de Ibagué, and also the Integrative Crop Ecophysiology group in the University of Barcelona. Okay, let's begin. This is our outline. First of all, we are going to talk about RGB imagery, RGB vegetation indexes, and also ideas about deep learning. Then we are going to talk about plant material, image acquisition, and then image resolution, image resize resolution. At the end, we are going to talk about image processing techniques using our very deep super resolution strategy, and also the relationship between vegetation indexes. Then we are going to talk about our results, and finally, conclusion and future works. Regarding RGB sensors, or therefore RGB imagery, I think it's important to say that this kind of images has become in one of the main proximal remote sensing approaches for crop monitoring and also for plant phenotyping application. Although the fact the RGB information don't necessarily capture all the essential data of the plant. I mean, we also have very important information in ultraviolet or near and long infrared information. Perhaps this is made due to its user-friendly nature, accessibility, relatively low cost, also high resolution, and I think it's important also highlight the availability of software and techniques that use image processing systems using RGB information. Perhaps this is the most common and versatile tool for remote sensing in application, for application in agriculture in nowadays. This is our next step. When we already have the images acquired using a conventional sensor, in this case an RGB sensor, we would like to acquire or calculate information related with the plant. Our idea is to have numbers or estimation related with what is happening in our crop. In this case, we use different kind of RGB vegetation indexes. In this case, we use the NGRDA index to estimate the nutrient status and biomass of the plant, and also the TGA to estimate chlorophyll concentration, and also in the end, we use the UVA, GA, and GGA indexes to estimate the photosynthetic area of the canopy. Our main idea is to calculate or estimate using numbers and specific information of the plant with the idea to correlate this information with a specific data. In this case, our specific goal is correlate this information with grain yield. This is the deep learning strategy that allows computational models to learn from data. This strategy has been used in a wide range of processes, including, for instance, agriculture application, such as grain yield or grain yield prediction. This is the specific application we are using in this work. The idea is correlate the data that we already calculate using vegetation indexes and correlate this information with grain yield. Of course, we have more application in agriculture, for instance, disease recognition or weed detection. In this specific case, we are using a very deep resolution, very deep super resolution architecture in order to increase the resolution of our input images. In our case, we have in this figure a low resolution input image, and the idea here is enter this information in the very deep super resolution architecture in order to increase the resolution of all of those images, with the idea to finally obtain as an output a very high resolution image. This experiment was carried out in Valladolid at the Itacil experimental station using 23 varieties of durum wheat with ounce, also using 3 replicates and 1 growing condition, in this case, the irrigated condition. This is the image acquisition step. Here, we decide to just walking around the plot, taking images using an RGB conventional camera with the idea to obtain this kind of zenithal images. When we took these images, we always use the same focal length and almost the same distance between the canopy and the camera. Also important to mention, we have 23 different varieties of wheat and also 3 replicates. In that way, we took 1 picture per replicate and 1 picture per variety. Okay, we already have the images acquired in the field, and now it's time to start the image resize simulation step. Here, we use the average pixel values without using any interpolation technique. In the figure, you can see in A, the original image size, and in B, the original image divided by 2. Comparing the figure A and the figure B, it's not clear to see the all differences, but at the end, in the figure F, you can see the image divided by 32. For that reason, it's easier to see the big spatial information differences in B compared with the original image B. Regarding the image size, the original image has around 3,000 pixels times 2,000 pixels, and then when we divide by 2, the image B change its size to around 1,500 times 1,000 pixels. And at the end, in the figure B, or in the step F, we have the lowest image size, which is 94 times 64 pixels. You can see the big differences between the first figure, the figure A, the high resolution image, and the figure F, the high resolution, the lowest resolution images. Here, our idea is to simulate lowest or highest distance, for instance, when you acquire images from drones. Okay, at this step, we already have the images with higher or lower resolution information. Now it's time to start with the image processing. In our case, we are going to use a super resolution strategy, which is a process to create high resolution images from low resolution images. The goal is to recover the high resolution information from one low resolution image. In this case, for this work, we are going to use the very deep super resolution network, which is a convolutional neural network designed to improve the image resolution from low to high information, adding the high frequency details of the image. This network employs the residual learning strategy, and the network learns how to estimate the residual image which contain information from the high frequency information details of the image. This network detects the residual image from the luminance channels of the image, which represent the brightness of each pixels. In addition, use the chrominance channel of the image that represent the color difference information. Also, it's important to mention that this network is trained using only the luminance channels because the human perception is more sensitive to the challenge in brightness than the changes in color. Here we have an example of the very deep super resolution architecture, and in the left side you can see the images that we acquired in the field, and in the right side you will see the images that we process using the very deep super resolution strategy. Here in this slide, we have a zoom of the previous figure, but only using the figure A and the figure B. And the idea here was to see in more details the results of process, the input image, the low resolution input image using the very deep super resolution strategy. As you can see, I hope you can see through the video, the differences in high resolution information related with the figure A and the figure B. You can see perhaps here in this part of the image that the frequency information is actually increasing. That is the specific idea that we use this image processing technique in order to increase the information of the image, and also, or specifically when we have lower resolution information. Here in this slide, we have more or less the complete idea that we use in this work. We use all of our database where we have low resolution information, which is these images, and then we also calculate all of the high resolution images using the very deep super resolution strategy. With these two databases, we calculate the complete indexes or RGB indexes in the complete image data. With this information, we obtain different kind of numbers depending of the indexes. For instance, GA and GGA give me back information related with the chlorophyll concentration, another important information, and then using the other indexes, I can obtain the information related with the photosynthetic area of the canopy, for instance. With this information, we actually have a big variety of numbers, and our idea is compare what is happening with these numbers. If we have a good correlation between each others, of if we have a different kind of results from the point of view of statistical analysis. Here we have a summary of our results, and our idea here is compare the Pearson correlation throughout the different indexes that we use, also divided by 2 and 4 and the different scales that we use for this study. In this case, we use the Pearson correlation between vegetation indexes that we represent in blue color as a low resolution images, and also the low resolution images processes, but the very deep super resolution strategy here in this figure in pink color. All of this information are related or correlated against grain yield. At the end, in the color green, we have the mean values throughout the different size that we use, and also using the different vegetation indexes that we calculate in this work. In general, we can see, for instance, here, when we divide by 2, that the Pearson correlation for the low resolution images and the Pearson correlation of the low resolution images processed by the very deep super resolution strategy is almost the same. And the same behavior is observed in the different samples or the different results that we can obtain in this work. For that reason, at the end, when we divide the different values and also when we observe the mean values in green color, we can see that the mean and also the standard deviation are really close of the values. In that way, also, we calculate the significant differences between the Pearson correlation of the values in blue color and the values in pink color, and we cannot observe significant differences between the Pearson correlation that we can get from the vegetation indexes and the yield or the grain yield that we obtain from the field. In that way, we we cannot say that the very deep resolution strategy is working well in the way that increase the grain yield information or the grain yield assessment in our study. In order to have more information related with the images, we also calculate the peak signal to ratio noise and the structural similarity index. We can see here in the result that the peak signal to noise ratio for the low resolution images are very close compared with the peak signal to noise ratio of the very deep super resolution strategy. The values are really close each other's, although we have lower values in the very deep super resolution strategy. Also, for the structural similarity indexes, we also have a very close results compared with low resolution and very deep super resolution strategy. Here also, we calculate if we have significant differences, and these differences were not found between the peak signal to noise ratio and also for the structural similarity indexes in these two values. Finally, as a conclusion and future work, we can say that although better visual resolution characteristics were observed when we use the very deep super resolution strategy and the low resolution images, the correlation of vegetation indexes against grain yield did not improve. This is important because at the beginning we thought that if we increase the high resolution information in the images, perhaps we could increase the grain yield assessment. Consequently, the significant differences between resolution and also between the use of low resolution information or higher resolution information using the very deep resolution information were not observed. These significant differences were not observed in this work. Perhaps this is made due to the convolutional network architecture that we use has been designed for image restoration, also for denoising and compression. This is working well for automatic recognition, but in our case, the results shows that there is no significant differences. Therefore, luminance and chrominance information may have been trained to improve only the visual characteristics instead of color feature. Perhaps as a future work, we can use the chromatic information or chromatic information channels of the image to increase or to improve the training information of the convolutional network in order to have better color feature to try to increase the grain yield assessment using vegetation indexes and also, of course, using RGB conventional camera information. Okay, this is the end. Thank you all for attending this presentation in this virtual symposium implying omics science. And this is my email if perhaps you have a question or perhaps we can work together. For me, it will be nice. And I hope we can see you in the next symposium. Thank you very much and goodbye.
Thank you, Jose. Very interesting talk. Congratulations on the work. I have some questions mostly associated with your conclusions, and that's regarding the use of color information. So humans and human vision in general has something called constancies. One of them is color constancies and motion constancies. We perceive a green tonality or a green hue at the same value independent of the amount of photons or light in the environment. Of course, up to a certain threshold. But cameras, on the other hand, and even multispectral cameras have a significant deviation in color coding when light changes, when you have, you know, luminance, and when you have intensities change in the image. How do you propose to compensate or normalize for those constancies in an unstructured environment? I mean, this is outdoors. This is using you know, imagery that requires somehow normalization prior to using it for training your CNNs, for example. And so that's my first question.
Yes, thank you for your question. You are talking about a key factor specifically because, of course, for us it's very important to use the luminance of the camera. But it's strange when we calculate all of indexes, they use specifically the luminance of the camera. For that reason, we thought if we change or we try to train in all of this architecture, the CNN architecture, we're going to increase the information, which is strange, a bit strange, because the luminance is also important. But in this result, we can see that it's most important in that way. In the way of indexes, it's most important the chrominance information. And also the other important point that you're talking about is about that we normally use all of this information in the field. Of course, the luminance is also important, but the chrominance must be included into the model.
That's exactly my point. Yes.
Exactly. The luminance is important, of course, but the chrominance also is important here.
I think the chrominance is actually more critical because you have, well, in an unstructured environment, you have a huge, you know, variation in chrominance. Most
light
likely
changes, I guess.
Yes.
Yes, of course. But actually, the indexes use as a very important point the luminance. But for that reason, we need starting to use luminance. Of course, chrominance is also important, but for sure, if we would like to have better results, we have to put into the model the chrominance information, of course.
Okay, great, got it. Well, again, great talk. I don't know if we have any additional pressing questions to the speaker at this point. If not, I'd like to thank Professor Fernandez again, virtual applause, and thank everybody else for a very productive and interesting session today. Tomorrow, a couple of announcements. We will have two keynote speakers, one in the morning, Professor Valente. He's going to be talking about the use of drones in phenotyping of crops. And near the end of the morning, we'll have Professor Gao talk about biosensing. Although his work is mostly related to humans, we're entering a collaboration now to look at biosensing in plants using the same technique he's using for humans. So right now he's using sweat as a way to reach the biomarkers in humans, so he can induce localized sweating, and we think we can do the same with plants, and that's very interesting. So tomorrow we will have additional peer review talks related to phenotyping. So I invite you all to be present, especially during the keynote talks. I would like very much for everybody to ask pressing and very, you know, poignant questions to the speakers, because that's the whole point of this symposium. I think the more technical and deep and really well validated you know, efforts are what, you know, are really going to pay off in terms of improving the productivity of agriculture and in terms of improving the challenges that we have in terms of zero hunger worldwide. So thank you again all. Have a great rest of the day. Yes, Jose.
Can I make an invitation? Yes, very quick.
Sure, go ahead.
It's just, I'm going to put a link here. It's a special issue for remote sensing. It's just to invite you publish in this special
Great,
issue.
great. When is it due?
It's starting now until the next year.
Okay, perfect. Perfect.
Okay.
Yeah, I see you're a guest editor in there. Great.
Thank you so much.
Definitely, we will. So there's a suggestion from Professor Teran regarding the poster sessions, and I think it's a very sound suggestion. Is it possible to have an extended session to have time to visualize the posters, perhaps at 7:30 a.m. or between 12 and 12:30 p.m.? I think it's an excellent suggestion, Wilson. So we will schedule it probably for tomorrow, and we will try to fit it in tomorrow, if not the day after. On Wednesday we have scheduled another poster session between 10 and 10:30. So we're gonna try and extend the poster sessions, considering everybody must have had some trouble navigating the system. So appreciate the suggestion, and we'll open it up. Yes, we do have really good posters, as mentioned. So I invite everybody to take a look at them. We'll keep them on as they are right now, without the interactive session with the authors, but we'll open a specific schedule for interacting with the authors anyway. Thank you again, and have a great rest of the day. Congratulations, everybody, and keep up the good work. Terrific. Bye.
Thank you.